Hello and welcome to Unacademy, a one-stop destination for all English medium civil service aspirants. Welcome to the Conquer Prelims 2026 Phase 2, Complete Science and Technology Revision Marathon. Before I start the session, a very important announcement. Our lowest ever prize 17,999 is extended and will end tomorrow. This, along with the CSC Assure, makes it the right time to take the Unacademy subscription. And you can now enroll for the Unacademy Revision Test Series for just 1,999. And the test series includes 2,500 high-quality prelims questions, 22 full-length tests with OMR sheets, 8 subject-wise online tests, real exam simulation available both offline and online to estimate where you stand in the preparation. All this at just 1,999. And do enroll before it is late. Don't forget to use the code IAS English. And let us start the session now. Most importantly, where to get the PDF of this particular session. The link to download this PDF will be provided in the description below the video. Also will be available in our official Telegram channel. Let us continue with the one-shot revision marathon on science and technology. This revision I will be doing in 2 parts. The complete science and technology we can divide into 6 major subjects like biotechnology, nuclear technology, nanotechnology, information and communication technology, defense technology, etc. Out of these 6 subjects, in today's class we will be covering biotechnology, nuclear technology, and then information and communication technology in the part 1. In the part 2, I will be covering the other 3 subjects, that is nanotechnology, space technology, and then defense technology. So, in today's class, let us continue with the first 3 subjects of science and technology, that is biotechnology, nuclear technology, and information and communication technology. This class may be for a duration of around 6 to 7 hours, and in this time I will try to cover almost everything that I cover in our comprehensive courses in a short duration. And I will try to minimize the breaks so as to give you more information possible, the maximum amount of the information possible. So let us continue with the session. Firstly, biotechnology. Okay. Hope my audio video is clear. You're able to hear me? Avinash, running notes is not needed because whatever I speak will be available in the printed material. You can just download the PDF from the description below or from our official Telegram channel. No need of taking any running notes. Okay, is my audio video clear? Perfect. Thank you. Let us quickly continue with the first subject, biotechnology. Starting with the cell. What is the cell? Cells are the building blocks of our life. Just the way the wall is constructed with the bricks, our body is constructed with the cells. It's not like we have a few cells in our body itself is cells. And every cell is covered by a membrane called as, yes, all of you, every cell is covered by a membrane. That membrane is called as plasma membrane or cell membrane. Plasma membrane or the cell membrane. Inside the cell there is a liquid or a fluid called as cytoplasm. Inside the cell there is a fluid called as cytoplasm. Inside the cytoplasm we have a lot of organelles. The most important organelle being the nucleus. Nucleus is the most important of all the organelles. And what all we are going to study in biotechnology will be happening inside the nucleus. It means we'll be discussing about DNA, chromosomes and all. Where do we find these DNA chromosomes? They are found inside the nucleus. Right? Then, now let us try to understand what is the DNA. Okay. DNA is made up of, inside our nucleus, we have 5 nitrogenous bases. Nitrogenous bases means what? Nitrogen containing basic chemicals. Nitrogen containing basic chemicals will be there. Okay. What are they? They are called as, their names are adenine, thymine, guanine, cytosine, uracil. Nothing to explain, they are just the names, memorize the names. Okay. Adenine, thymine, guanine, cytosine, uracil. What are they? All the 5 are nitrogen containing basic chemicals. Now, any one nitrogenous base, for example, one adenine. To that, if I add one sugar, then this complex is called as this base plus sugar. This base plus sugar is called as nucleoside. Then, to that nucleoside, that is the base, like for example adenine plus sugar. This is called nucleoside. To this nucleoside, if we add one phosphate, nucleoside plus phosphate together is called as a nucleotide. Means a nucleotide contains 3 components in it. A nucleotide contains one base, that can be adenine or thymine or guanine or cytosine or uracil, any one base, plus one sugar plus one phosphate together is called as nucleotide. Only these two together is called as nucleoside. Okay. So, if you observe, just look at here and keep answering. What are these called as? Adenine, uracil, guanine, cytosine, adenine. What are these called as? They are bases. They are bases. Then here there is one name called R. R stands for what? R means ribose. What is ribose? It is the name of a sugar. Like glucose, fructose, like that, this is also one sugar. Now, okay. Now look at here. You have the base plus sugar. So this structure can be called as what? These two together can be called as what? Just the ribose sugar plus adenine, nucleoside. To the nucleoside, a phosphate is added. Like for example, what is this called as? This complete ball. This is base. This is sugar. This is phosphate. This complete structure is called as the nucleotide. Then next level what happens? All the nucleotides, 1, 2, 3, 4, 5, you can see 5 nucleotides here. The chain of nucleotides is called as nucleic acid. The chain of nucleotides is called as nucleic acid. This is one nucleic acid. Okay. Then, so now we have 2 types of nucleic acids. One is DNA and the other one is RNA. Is it clear? One is DNA, the other one is RNA. Let us see the differences between the two. In the DNA you have ATGC. Tell me which nucleotide is missing in the DNA? Uracil is missing. Whereas RNA contains AUGC. Which nucleotide is missing? Thymine is missing. The second difference is in the sugar. Which sugar is present in the DNA? Deoxyribose sugar. Which sugar is present in the RNA? Ribose sugar. So ribonucleic acid means what? Ribose sugar containing nucleic acid. Nucleic acid containing the ribose sugar. Deoxyribonucleic acid means what? Deoxyribose sugar containing nucleic acid. The nucleic acid with the deoxyribose sugar. The third difference is DNA is double stranded. It means there will be two chains of nucleotides on this. Two chains of nucleotides will be present. Whereas RNA is single stranded. Means what? There is only one chain of nucleotides on this. There is only one chain of nucleotides on this. Let us try to understand the same differences with the help of this diagram. Okay, look at here all of you and answer this question. Is this nucleic acid an RNA or a DNA? It is an RNA. Okay, why? Because it is written RNA here, ma'am. Okay, fine. That is not the only reason. If you observe here, you have A, U, you have uracil. So it is not RNA. It is, it is not DNA. It is RNA. Uracil is present. Second point to note, you have ribose sugar, not deoxyribose sugar. It's a ribonucleic acid. Second point to be noted. Third point to be noted, there is only one chain of nucleotides, single stranded. Now look at the DNA. Here you have, note down this point, this is thymine. Thymine is present. Point number 1. DNA will have ATGC, not uracil. Okay. Point number 2. You have two chains of nucleotides. You have two chains of nucleotides. Second point. The third point, you have deoxyribose sugar. Deoxyribose sugar. Therefore, it is DNA. Okay. And which is our genetic material between RNA and DNA? DNA is our genetic material. DNA is our genetic material. What do you mean by DNA is our genetic material? The DNA determines or decides all our characters. The DNA determines or decides all our characteristics. Therefore, we call it as the genetic material. And what do you mean by this pink color lines? That is called as base pairing. There is a mechanism called base pairing. Simple. We have 5 nitrogenous bases, ATGCU. Base pair means what? A pair of means 2 of the bases. In the base pairing rule, adenine will always pair with thymine. It means thymine will always pair with adenine. Okay. Where? If it is in the DNA. But do you have thymine in the RNA? No. So if it is RNA, then the adenine will pair up with the uracil. So AT or AU. Then guanine always pair with cytosine. Let it be DNA or RNA. Because you have guanine and cytosine both in the DNA and RNA. Is it clear? And the bonds between these bases are called as hydrogen bonds. The bonds between these bases are called hydrogen bonds. And there are 2 hydrogen bonds between AT and 3 hydrogen bonds between GC. Their chemical structure is like that. There is no deep logic behind that. Okay. So this is the DNA structure. With this we understood the DNA structure. After we understand what we need, what is chromatin and chromosomes. If you compare DNA as one thread, if you compare the DNA as one thread, let me just take a wire. Right. Am I visible? Okay. So if we compare DNA with a thread, this is like DNA. This is like DNA, right? Then this DNA will get coiled like this. Okay. Now what is this? This is also DNA only, but condensed DNA. That condensed DNA is called as chromatin. That condensed DNA is called chromatin. But that is also DNA only. Observe it. Okay. Now what happens? This DNA will get more tightly condensed. This DNA will get more tightly condensed. That maximum condensed DNA is called chromatid. Okay. So what is this? This is DNA but loose. What is this? This is also DNA but tight. Then it is called chromatin. Okay. Now what is this? This is also DNA but tightest. Then what we call it as chromatid. Is it clear? Then two chromatids together is called as one chromosome. What is a chromatid? Look at here. This structure is a chromatid. This is one chromatid. Don't count it as 1, 2, 3, 4. No, you don't have 4 chromatids over there. You have only 2 chromatids. Chromatid looks like this. A chromatid looks like 8 shape. This is one chromatid. This is one chromatid. Okay. So I repeat, DNA when it is loose, it is called DNA. A condensed DNA is called chromatin. Maximum tightly condensed DNA is called chromatid. Two chromatids together is called as one chromosome. A chromatid looks like this. A chromosome looks like this. Okay. Now tell me, one chromatid contains how many DNA molecules? Look at here. One chromatid contains how many DNA molecules? One chromatid contains just one DNA, not 2 DNA. There are 2 strands, but only one DNA. It is one DNA, but 2 strands. Don't confuse between 2 strands and one DNA. Okay. Look at here carefully. This is one chromatid. Divide like this. This is one chromatid. One chromatid contains one chromatin. One chromatin contains one double-stranded DNA. Is that clear? So one chromosome contains 2 DNA molecules. One chromosome contains 2 DNA molecules. But both are identical. Why both are identical? Because they are the replicated copies of each other. They are the replicated copies of each other. It means if this DNA replicate, you will get the other DNA like a Xerox. This is like a Xerox to the original one. Okay. So these 2 DNA molecules are identical to each other. Okay. That is chromosome. Now, in our body, each cell contains, every single cell contains 23 pairs of chromosomes. Means we have 46 chromosomes, not in our complete body. 46 chromosomes in each cell. Is it clear? In this first 22 pairs of chromosomes are called autosomes. What are autosomes? Autosomes are nothing but chromosomes only. Autosomes are nothing but chromosomes only. But the first 44 chromosomes are called autosomes. Okay. What is the duty of these autosomes? They control or decide or determine the characters that are common for both the sexes. Like for example, you have liver, heart, skin, blood, bones. In this, there is no difference between male and female. Male and female difference lies only in the reproductive system. So leaving the reproductive aspects, general aspects like we all have 2 kidneys, 1 liver, such characteristics are controlled by the autosomes. The 23rd pair of chromosomes, okay, please notice this. Not all pairs, only the 23rd pair of chromosomes are called allosomes. What do you mean by allosomes? Allosomes are also chromosomes only, but the last pair of chromosomes are called allosomes. The last pair of chromosomes are called as allosomes. Is it clear? Okay. Right. The last pair of chromosomes is called as allosomes, right? Then, and what are allosomes? Allosomes are also called as sex chromosomes. Okay. Why are they called sex chromosomes? Because they decide the sex of the person. They are called sex chromosomes because they decide the sex of the person. Is it clear? Okay. And no 700, it is not 23 allosomes. Only 23rd pair is allosomes. First 22 pairs like this. Look at this diagram. Okay. Imagine what is this? This is first pair. Is this clear? This is first pair. Chromosome number 1. Again chromosome number 1. This is second pair. Then I cannot draw 23 pairs. So I just kept dot. So imagine 3 to 20 in between, third pair, fourth pair, 10th pair, 12th pair, like that. This is 21st pair, 22nd pair. And what do you mean by a pair of chromosomes? Look at here. What is this? This is a chromatid. A chromatid looks like 8 shape. Then what is this? It's a chromosome. A chromosome looks like this. Then a pair of chromosomes will include how many chromatids? One chromosome contains 2 chromatids. So a pair of chromosomes include how many chromatids? Everybody? A pair of chromosomes means 4 chromatids. Okay. Now look at here and tell me. I am circling something here. Is this one chromatid or one chromosome or a pair of chromosomes? What I have circled here, is it one chromatid or one chromosome or a pair of chromosomes? It's a pair of chromosomes. Like this, how many pairs you have? Like this, we have 23 pairs. So first 22 pairs, it means 44 out of total 46 chromosomes are autosomes. They are not allosomes. Is it clear? Only last pair, that is only 2 chromosomes out of total 46 are called as allosomes. Allosomes are also called as sex chromosomes. And these 2 chromosomes are which pair? Only the 23rd pair. Other pairs are autosomes. Okay, so to give you clarity, in this diagram, I have shown autosomes in yellow color and allosomes in pink and blue color. Is it clear? Right. If this is clear, all of you tell me, in the males, okay, fine. I told you that 23rd pair is allosomes. Then, the allosomes are also called as sex chromosomes, right? Allosomes are also called as sex chromosomes. We have 2 sexes, male, female. So we need 2 sex chromosomes also. Okay, we have 2 sex chromosomes, X and Y. In this, X chromosome is called female sex chromosome. So all of you tell me, what is X chromosome called as? What is X chromosome called as? Female sex chromosome. Why this is called female sex chromosome? Because it controls all female sex related characteristics. For example, egg production, ovaries, uterus, all female reproductive system related characteristics are under the control of this chromosome. Therefore, it is called as female sex chromosome. Y chromosome is called as male sex chromosome because it controls the male sex characteristics. Testis, sperm production, testosterone hormone, The total male reproductive system is under the control of this chromosome. Therefore, it is called male sex chromosome. Is it clear? Now, focus on this point. The 23rd pair in the females is XX. The 23rd pair in the males is XY. All of you check these 2 points. The 23rd pair in the females is XX. The 23rd pair in the males is XY. Okay. Now tell me, is this a male or a female in this diagram? Look at this diagram and tell me, is it a male or a female? Is it the diagram of a male or a female? The diagram shows a male because 23rd pair contains one X chromosome and one Y chromosome, XY. Okay. 23rd pair in the male is XY. 22nd pair in the male is, okay, tell me 22nd pair in the male is XY or XX? Everybody please. The 22nd pair in the male is XX or XY? Okay. So the question itself is wrong. 22nd pair in the male is neither XX nor XY. Then what it is? It is an autosomes. But they are autosomes, that is 22, that's all. This 22nd pair, this is neither an X chromosome nor a Y chromosome. Then what chromosome is it? It is an autosome. More specifically, which autosome? 22nd chromosome. Okay, it has no name. You have only number. Okay, only number. Is that clear? Okay, so now tell me, the 5th pair in the females is XX or XY? Female. 5th pair in the female is XX or XY? All of you please. Neither XX nor XY. It is an autosome, more appropriately 5 and 5. So remember this point. Now, moving on to the next term, that is ploid. First memorize this word ploid. Text me that term once. Ploid. Ploid means one set. Set means like one whole package. Like one whole bundle. Okay. Please remember, not pair. I am not speaking about pair. Okay. Pair means 2 chromosomes. That is different. One whole bundle, one whole package, one whole set of all chromosomes in a cell is called as a ploid. If that is the definition, one ploid is equal to how many chromosomes? In one package, in one bundle, in one complete set, how many chromosomes will be there? Yes, everybody? 20, 23 or 46? One ploid is equal to 23 chromosomes because it is only one set, not 2 sets. Therefore, it is not 46, only one set, okay, of all chromosomes. Only one set of all chromosomes in a cell. Means ploid is equal to 23. We represent the word ploid with the letter N. N is equal to 23. N is equal to 23. Is it clear? N is equal to 23. And how many chromosomes we have? We have 46 chromosomes. Means look at here. This is one ploid and this is another ploid. Okay. So, if one ploid is equal to 23, if one ploid is equal to 23, then 2 ploids is equal to how many chromosomes? 2 ploids is equal to 46 chromosomes. Okay. Means we are diploid. Agreed? Di means 2. 2 ploids in each cell. We are diploid means we have 2 ploids in each and every cell. Okay. Diploid means 2 into 23, 46 chromosomes we have. Is that clear? Then in that case a triploid will have how many chromosomes? 3 into 23. Tetraploid will have 4 into 23. Okay. Hexaploid means 6 copies, 6n means 6 into 23. Okay. Heptaploid means 7n, means 7 into 23. 7 into 23. But is it 23 always? No. Okay. Note down a few points. In humans, we have 23 chromosomes. In Drosophila, that is fruit fly, you have 4 chromosomes. In maize, okay, that is corn, you have 10 chromosomes. You have 10 chromosomes. Then in pea plant, okay, we have 7 chromosomes. So now can you tell me, diploid, a 2n fruit fly, that is Drosophila, will have how many chromosomes? 2 into 23, 2 into 7, 2 into 10, 2 into 4. 2 into 4, that is 8 chromosomes. Whereas a 2n human being will have 2 into 23, that is 46 chromosomes. Okay. So n means it need not always be 23 chromosomes. It varies from one species to the other species. Different species have different number of chromosomes, whereas the 2n, 3n, 4n, they speak only about number of copies. Whereas these numbers are the number of chromosomes. These are the numbers of chromosomes. Is that clear? Okay. Then now look at here. As I told you, this is one pair of chromosomes, second pair, and I cannot draw 23 pairs, so therefore dot. This is 23rd pair. In that case, look at here, all these together, this complete package is called one ploid, one set of all chromosomes in a cell, 23 chromosomes. This comes from the mother. It means we get 23 chromosomes from the mother. And the other 23 chromosomes, the other package, okay, will come from the father. It means what? We have each chromosome in 2 copies. Is it clear? Each chromosome in 2 copies. One copy from mother. The second copy from father. Same way we also have 2 ploids. So one ploid comes from the, it means 23 chromosomes, comes from the mother. And the other ploid comes from the father. It means, do we have 46 different chromosomes? No, we don't have 46 different chromosomes. We have only 23 chromosomes. We have only 23 chromosomes, but each chromosome in 2 copies. We have only 23 chromosomes. We don't have 46 chromosomes. But each chromosome we have in 2 copies. So if you just look at this diagram, look at this. This is first chromosome. What is this? This is also first chromosome only. Other copy of first chromosome. Therefore, we don't say that we have 46 chromosomes. We say that we have 23 pairs of chromosomes. Why don't we call it as 46 chromosomes? Because we don't have 46 different chromosomes. We have only 23 different chromosomes and we have them in 2 copies each. Is that clear? So, now that is about the word ploid. Now, as I told you, if you have 3 ploids, then it is called triploid, tetraploid, pentaploid, hexaploid, heptaploid, octaploid, nonaploid, like that. What is a common word I can use for all of them? Triploid, tetraploid, pentaploid, hexaploid, heptaploid, octaploid, nonaploid. I want one common word that will cover all of them. What we can call them as? We can call them as polyploids. We can call them as polyploids. Do we have any polyploid human beings? Never, never in the medical history. Okay? Not only human beings, humans and other animals cannot tolerate polyploidy because these chromosome errors, it means I am supposed to have 46, by mistake if I have 69 chromosomes, okay, by mistake if I have 69 chromosomes, then what happens? It will disrupt the sexual reproduction. So therefore we cannot survive. Whereas plants can easily tolerate polyploids because they don't have only sexual reproduction. They also reproduce through asexual reproduction, in which chromosomes do not play an important role. Is it clear? Look at this. Plants also reproduce through asexual reproduction, in which chromosomes do not play important role. Okay, so chromosome errors do not kill the plant, but chromosome errors kills an animal or a human being. So polyploids cannot be tolerated by humans or other animals, but polyploids can be easily tolerated by the plants. By the way, why do we make polyploids? Look at here. A diploid strawberry looks like this. The strawberries that are available in the market are octaploid strawberries. It means 8N. Is it clear? They have each chromosome. The strawberries that we get in the market, they have each chromosome in 8 copies. They have each chromosome in 8 copies. Is it clear? And why do we make polyploids? Polyploids can increase the size or increase the flavor or increase the protein content. It means polyploid, making a plant polyploid do not give it any new characteristic, but whatever characteristics are already there, their quantity can be enhanced. If it is already red color, you will get bright red color. If it is already sweet, you will get sweeter taste. If it is small in size, you will get bigger size fruits. That is the reason why we make polyploids in the plants. Is the polyploidy concept clear? Okay. Now let us see chromosomal aberrations. Chromosomal aberrations means mistakes in the chromosomes. Okay. Now tell me a few questions, all of you, very quickly. We all have 46 chromosomes. So answer these questions. XX is a normal, tell me, normal male or female? XX is a normal female. Then XXX should be super female syndrome. Super female syndrome. Okay? Right. Then XY is a normal male. Whereas XYY means already a male. If a male contain one extra male chromosome, then it is called as super male syndrome. Super male syndrome. Then X0, it means they don't have 2 X chromosomes in the last pair. Look at here. This is missing. Okay, there is a blank here. Did you notice? X0. That is called as Turner's syndrome. Turner is a scientist who gave that name. Therefore, it is called Turner's syndrome. Same way XXY. Okay, in the last slot, if you have XXY, that is called Klinefelter's syndrome. Klinefelter's syndrome. Klinefelter is the name of the scientist. Same way, XXXY. It is called extreme Klinefelter's syndrome. Extreme Klinefelter's syndrome. Okay. Now, a few questions. All of you answer these questions, please. Okay. Tell me the total number of chromosomes in each case. What is the total number of chromosomes in a normal female? First, 46. What is the total number of chromosomes in a super female? 47. Okay. If any of you did not understand how 47, look at here. Okay. This is super female syndrome. Super female syndrome. Right? Then, trisomy of X chromosome. Trisomy. Look at this word. Tri means what? 3. Somy means what? Chromosome. Okay. Trisomy of X chromosome means what? You have 3 copies of X chromosome here. X, 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 X. That is called trisomy. Then what will be the total number of chromosomes? 46 here plus 1 extra X chromosome. Look at here. This is 46 and this is 1 extra X chromosome. Therefore, it is called as trisomy of the X chromosome, super female. Then you will have 7 chromosomes. Okay. Then a normal male will have 46 chromosomes. There is a super male XYY. Look at the super male XYY. With this, it is 46 chromosome plus 1 extra 47 chromosomes. Is it clear? 47. Turner's syndrome will have how many chromosomes? 45. Look at here. Okay, only 45. 46th chromosome. 46th one is missing. 46th one is missing. So only 45. XXY. Look at here. This is called Klinefelter's syndrome. Klinefelter's syndrome. XXY. Okay. With this, it will become 46 plus this is extra plus 1. Total 47 chromosomes will be there in this. 47 chromosomes will be there. Okay. Whereas extreme Klinefelter's will have 48 chromosomes because with XX it will become 46. Now plus 2 chromosomes. It will become total 48 chromosomes. Very good. Thank you so much for giving all the right answers. Okay. Now just continue to answer. Okay. Guess what will be the sex in each case. For example, XX. Is it a male or a female? XX, is it a male or a female? It is a female. Then triple X also will be a female because XX is already a female. In a female, if you have one extra X chromosome, obviously it will be female only. Then XY is a male, we already know. Then XYY will be, of course, a male because XY is already a male. In a male, if there is an extra male chromosome, obviously it will be male only. Then X0 is a female. Why? Why this is a female? Because simple. Which chromosome gives male characteristics? Y chromosome. Do they have Y chromosome here? No. Without Y chromosome, how can she get male characteristics? No male characteristics. Therefore, it is female. Does not mean that it is a healthy female. Okay, infertile, abnormal. But on the whole, female only, not a male. Is it clear? Klinefelter's is a, just guess and tell me. Klinefelter's is a male or a female? Some students say that you have 2 female chromosomes and only 1 male chromosome. So the net result is it should be female, but it is wrong. Reason is Y chromosome can dominate. Y chromosome can dominate the X chromosome. That is the reason why. Okay, if just one Y is present, if just one Y is present, okay, even if you have 3X chromosomes, okay, then the gender will be, the sex will be male only. Okay, you have 1X chromosome, 2X chromosomes, 3X chromosomes, 1X chromosome like this in the case of a normal male. Okay, in the normal male also Y is dominating the X. That is the reason why a male do not exhibit female characteristics. Okay, in the Klinefelters also you have 2X chromosomes and 1 Y, but Y is dominating both of them. Here you have 3X chromosomes and 1 Y, but that Y is dominating all the 3 chromosomes. So in these two cases also, the overall sex will be male. Overall sex will be male. Klinefelters is a male because Y chromosome, okay, Klinefelters is a male because Y chromosome dominates the X chromosome. Even if you have 3 female chromosomes, just 1 male chromosome, still this 1 male chromosome can dominate the 3 female chromosomes, making the overall sex a male. Is that clear? Okay. Now that we understood the features, let us now understand some symptoms of this syndromes. Turner's syndrome, it is called monosomy of the X chromosome. Why monosomy? Okay, look at here. The word mono, okay, the word mono means one. Mono means one. Somy means chromosome. So one chromosome, you have only one chromosome here. And symptoms, they are female only because Y chromosome is missing, but they have short stature, means height will be very short, and they have some mental retardation possibility. But socially, we avoid using this word mental retardation because it can demean or demotivate or disrespect some people. So therefore, we usually call her or him as a special child. Okay, but to explain the concept, I had to use the term. Is it clear? These are the symptoms of the Turner syndrome. Are the symptoms clear for all of you? Okay, next, Klinefelter's syndrome. Trisomy XXY. Gender is male because Y chromosome can dominate on the X chromosomes. Symptoms are male but infertile man, not a fertile man. Okay, sperm count will be lesser, cannot produce as many sperms as a normal male would produce, or no sperm production at all, either 0 sperms or lesser number of the sperms. Okay, they also exhibit some feminine characters. It can be in the walking, it can be in the bodily appearance or breast development or voice like a female. So on the whole male, but will exhibit some feminine characteristics. Okay, this is called Klinefelter's syndrome. Next you have super male syndrome. This is also trisomy. Which trisomy? Tri means 3, somy means chromosome. 3 chromosomes are there. Which 3 chromosomes are there? XYY. XYY will be there. Is that clear? Then gender will be male. Okay, symptoms are they are taller than the average male. They have learning problems and they have criminal bent of mind. It means they have a tendency to do the crimes, but it is not a proven fact. It is just observed in some studies. It means in the prisoners there is a slightly higher frequency of the super male patients. So with this, because of that reason, they kept it as one of the symptoms. It does not mean that every super male is a criminal or every criminal is a super male. Okay, super males have some tendency to do the crimes. That's all. Okay, that too just suggested in a few tests, not like a proven fact. Next, super female. This is also trisomy. That means 3 chromosomes. Which 3 chromosomes? 3 X chromosomes will be there. Gender is female, no doubt there is no Y at all. So therefore the gender will be female only. Symptoms are they are taller than the average woman. They have normal levels of fertility. It means when you see a girl, you can't say whether she is a normal girl or a super female. Okay, no major problems. Looks quite normal. Can also produce the egg cells. They are fertile also. Rarely they have some learning difficulties. Learning difficulty means slightly autistic. Okay. Then comes the most important one, Down's syndrome. Till now what all we have seen, that is Turner's, Klinefelter, super male, super female, extreme Klinefelter. They are all sex chromosomal aberrations. whereas this is an autosomal aberration, some mistake in the autosomes. It is trisomy of the 21st chromosome. So look at here, 21st chromosome is present in 3 copies. A normal person should have 21st chromosome in how many copies? Yes, everybody? A normal person should have 21st chromosome in how many copies? 2. But the Down syndrome person will have autosomes in 21st chromosome in how many copies? 3. This is the most common chromosomal aberration. When compared to other chromosomal aberrations in the society, we get to see the Down syndrome patients more frequently. Okay. Symptoms include delayed development, learning disability, autism related problems. Autistic means behavioral problems, problems getting socialized and having lesser IQ levels. Okay. So I repeat, autism can have 3 different aspects. One is low IQ levels, 2 having problems in getting socialized. Okay. 3 cognitive skills will be impaired and they will have difficulty mingling with the people. Okay. So that kind of thing is called as like autism. Done. So with that we also completed the sex chromosomal aberrations. After that, let us move on to the what is egg cell, what is sperm cell, what is zygote, what is embryo, what is fertilization, how the sex of the baby is decided, who will decide the sex of the baby, identical twins. So these are our next topics lined up. Can we proceed? Okay. Yes, Avinash, there is a reason for the Down syndrome being more common, because if a chromosome is more important, okay, then if there is abnormality with respect to that chromosome, the child will not even survive till the birth. So therefore will get aborted. 21st chromosome is a very small chromosome, relatively less important. So 21st chromosome related abnormality, okay, cannot kill the baby while it is in the womb. So more such babies can take the birth. That is the reason. I hope it is clear, Avinash. Okay. Why all extra chromosomes add towards the right side? That is just because your Bindu madam is a right-hander. Okay. In our nucleus, the chromosomes are not arranged like this. They are all just pulled together. They don't line in the queues like this. Okay. Just because I am a right-hander, I have drawn it on the right side. In the chromosome, in the nucleus, for example, the first chromosome can be beside the 22nd chromosome. Like this, all chromosomes are just jumbled up. They don't line in the queues like this. Is that clear? Okay. How can there be 3 copies of the 21st chromosome? That is because of the non-disjunction of the 21st chromosome during either oogenesis or during the spermatogenesis, but don't worry, you don't need that. Okay. For a GS level, you don't need the reason how it came like that. Is it clear? That we will discuss in the optional subjects. You don't need it for the GS. Okay. Then now let us proceed with what is an egg cell, sperm, zygote, embryo, fertilization, who will decide the sex and all that. Okay. Now look at here and complete focus. What is this? This is a female somatic cell. Somatic cell means what? The word soma means body. So somatic cell means body cell. Somatic cell means body cell. Okay. Do we have sex chromosomes in the hair cell? Yes. Do I have X and Y chromosomes? I am a female, so do I have X and X chromosomes in my hair cell? Do we have sex chromosomes in the hair cell? Yes, yes, because I told you that we have each cell contain 23 pairs of chromosomes, if you remember that point. Each cell contain 23 pairs of chromosomes. So yes, I have sex chromosomes in the hair cell also. Then you must know, what are they doing there in your hair cell? They don't do anything, they are silent, but they are present. It means in every cell we have all the 23 pairs of chromosomes. Okay. So a female somatic cell, it means if I take my skin cell like this, that skin cell will have, okay, 46 chromosomes, 22, 22. What is that 22, 22? 22 plus 22, 44 autosomes plus 2 X chromosomes. How many chromosomes you can see in this circle? How many chromosomes you can see in this circle? 46 chromosomes, and the somatic cell is a diploid cell. The somatic cell is a diploid cell, 2n, total number of chromosomes 46, 22 plus 22, 44 autosomes plus 2 X chromosomes, that is 46 chromosomes will be there. Same way, if it is a male somatic cell, it will have again diploid, 46 chromosomes will be there, and the 46 chromosomes are 22, 22, 44 autosomes plus X and Y 2 chromosomes, okay. 44 autosomes plus X and Y 2, 46 chromosomes. Is it clear? Okay. Means which chromosomes are common between male and female? They are autosomes. Here also you have 44 autosomes, 44 autosomes. So 44 autosomes plus XX is a female, 44 autosomes, okay, plus XY is a male. Is that clear? But 44 autosomes are common for a male and a female. Now, okay, the egg cell contains only, okay, yes Naman, we can do. With the hair cell you can say whether it is a female hair or a male hair. We use it in the forensic testing. Okay, right. Now, look at here. The egg cell, what is this? Focus on this. Okay. No, chromosomal aberration is not diploid, mango. Okay, mango chromosomal aberration is not diploid. Diploid is normal. Any deviation from diploid is called chromosomal aberration. Okay. Now, so all of you tell me, a somatic cell contain total how many chromosomes? 46 chromosomes. Means it is a diploid cell. Diploid cell. Okay. Then the egg cell contain, look at here, this is the egg cell. An egg cell contain how many chromosomes? Yes, all of you, egg cell is a N. It will have only 23 chromosomes. So here I am using the word haploid. Note this down. Haploid. Haploid means half the number of ploids, right? Not half the ploid. Half the ploid means what? Half of 23 means 11 and half. Is there any meaning? No, wrong. It's not the definition. Half the number of ploids. So what does it mean? We have how many ploids? We have 2 ploids. We have 2 ploids. So half of them is 1 ploid. 1 ploid. Okay. So it means, can I say that the haploids of diploids are monoploids? Can we say like that? Haploids of diploids are monoploids. Means what? What does it mean? Haploids of diploids are monoploids means half of 2 is 1. That is the meaning. You have 2 ploids. Half of them means 1 ploid. Monoploid. So is our egg cell a haploid cell? Is a human egg cell a haploid cell? Yes, haploid cell. Yes, human egg cell is haploid. Okay, our eggs and sperms are haploid. Eggs and sperms are haploid. Okay, or monoploid. Why? Okay, so that when they fuse, the zygote and therefore the next generation can be diploid. Is it clear? Just imagine if the egg is diploid, sperm is diploid, then the zygote will be tetraploid. If the egg is diploid, sperm is diploid, zygote will be tetraploid. Is that okay? Can human beings tolerate tetraploidy? No. So therefore, if the egg is haploid, sperm is haploid, it means if the egg is monoploid, sperm is monoploid, then the zygote will be diploid. So that generation after generation, we can maintain to be diploids. That is the reason why in every generation the eggs and sperms will be haploid. It means egg will have only half the number of ploids when compared to your somatic cell. Is that clear? Egg and sperm will have only half the number of ploids compared to your somatic cell. Clear for all of you? Right. Okay. So now this is egg cell. Okay. These are the pink color ones, egg cells. Green color, blue color ones, sperm cells. What are they? Sperms are nothing but the male reproductive cells. Okay. Eggs are nothing but the female reproductive cells. Let us note down that definition also. Haploid, half the number of ploids. We have 2 ploids. So half of them is 1 ploid. Therefore, haploids of diploids are monoploids. Means half of 2 is 1, like that. Done? Now look at here. If the egg fuse with, okay, fine, tell me, how many types of egg cells you have here? How many types of egg cells you have here? Only 1 type. How many types of sperm cells you have here? 2 types. What are they? X type and Y type. If the egg fuse with X type sperm, then it will form 22 XX, that is female zygote. And if the egg fuse with Y type sperm, then it will form 22 XY, that is male zygote. Female zygote grows into a female baby, male zygote grows into the male baby. So in the human beings, who decide the sex of the person? Can the mother decide the sex of the baby or the father decides the sex of the baby? Actually, it is none of them. Neither the mother nor the father can decide the sex. It is purely random. So blaming somebody for giving birth to a female child is absolutely wrong. Okay. But if you have to blame somebody for giving birth to a female child, then it must be the male, because males have 2 types of the sperms. The sperm decides, okay, whether the baby is going to be a male or a female. Is it clear? So this is nothing but the zygote. Okay. Now the zygote will undergo a process called cell division. Cell division means one cell splits into 2. Then the 2 will become 4, 8, 16, 32, 64, 128, 256. That is how the cell number goes on increasing, and it will form a group of cells called as embryo. A group of cells called as embryo. What is an embryo? Undifferentiated mass of cells. Undifferentiated mass of cells means what? A group of similar cells. Undifferentiated just means not different. In that group all the cells look similar. In that group all the cells look similar. They don't have much difference. Undifferentiated mass of cells are a group of similar cells, that is called embryo. In the early stages it looks like a bunch of grapes, and gradually it looks like, okay, after some time it looks like a ball, but it will not look like a human being. It will not look like a baby. Is it clear? Okay. Clear till now? Then, so now the embryo, the cell division continues to occur and differentiation also occurs. Okay. What are the two mechanisms that occur in the embryo? What are the two mechanisms that occur in the embryo? Cell division. The cell number goes on increasing. Not only the cell number is increasing, also the cells will start becoming different gradually. It means gradually some cells will start becoming into liver. Some cells starts growing into pancreas. Some cells starts growing into the heart. So gradually the cells are differentiating. So which two mechanisms occur here? Embryo to fetus. We have two things. One, cell division. Two, differentiation. Embryo is undifferentiated. Fetus is completely differentiated. Embryo will become the fetus by two mechanisms. One is cell division and the cell differentiation. Okay. Then what is fetus? The baby which is not yet born, the baby which is still inside the womb is called as fetus. Then after the birth, the newborn baby is called neonate. The newborn baby is called neonate because neo means new, natal means birth, okay, or newborn. Is it clear? Okay, this is how the baby will be born. Now, in general, a female releases only one egg cell per month. Is it clear? A female releases only one egg cell per month. Therefore, twins are rare. Do you agree? A female releases only one egg cell per month. Therefore, the twins are rare. Right? Okay. But rarely sometimes if two eggs are released, then a man releases how many sperms? A man can release 500 million sperms. Is that clear? So by chance if a female releases two egg cells, is there a chance that two sperms can fuse with them? Yes. Then what happens? Two zygotes will be formed. The two zygotes will grow into two babies. Such babies are called as dizygotic twins because di means two. Two zygotes, okay, formed the twins. Therefore, they are called dizygotic twins. Is the name clear? Then, they are also called as non-identical twins because they do not look identical. Why don't they look identical? Because they are formed from two separate zygotes. They are not formed from the same zygote. So, therefore, they do not look identical. Okay, they don't look identical. So, do they look opposite like lambu jumbu, black and white, something like that? No, they can be similar. How similar they can be? They can be as similar as the normal siblings. They can be as similar as the normal siblings. Therefore, they are also called as fraternal twins. Fraternity means brotherhood. Even you have the word in the preamble also. Fraternity means brotherhood. Just like normal brothers and sisters, siblings like. Okay. Means genetically dizygotic twins are just like the normal siblings. Is it clear? The only difference is, look at here. If these two zygotes are formed on same day, then they are called dizygotic twins and born on the same day. But if these two zygotes are born, okay, if these two zygotes are formed with some 1 or 2 years gap, then they are called as normal siblings. So, do you agree that dizygotic twins are genetically just like normal siblings? Dizygotic twins are 700. I told you, usually female release only 1 egg cell per month. But there is a chance that a female can release up to 10 eggs also in 1 month. Okay? Most of the times only 1 egg. But more eggs are not impossible. A female can release more than 1 egg cell. Okay? Twins are rare because a female usually release only 1 egg cell. But twins are possible because many times a female can actually release more egg cells. If a female release, okay, if a female release 2 egg cells and if the 2 egg cells fuse with 2 sperms and forms 2 zygotes, they are like dizygotic twins. They are just like normal siblings. If these 2 zygotes are formed on the same day, the 2 babies are born on the same day, then they are called dizygotic twins. The same 2 zygotes are formed with 1 or 2 years gap, then they are called normal siblings. So genetically dizygotic twins are just like normal siblings. So based on this, now tell me, okay, can we have 1 male, 1 female dizygotic twins? Can we have 1 male, 1 female dizygotic twins? Yes? Yes, because they are just like normal siblings. For example, you have 2 egg cells. How many sperms you have? You have 500 million sperms. Means in this, roughly 250 million are X type. Roughly 250 million are Y type. If both egg cells, keep filling my sentences, if both egg cells fuse with X type sperm, if both egg cells fuse with X-type sperm, then 2 females will be born. If both egg cells fuse with Y-type sperm, then 2 male babies will be formed. If one egg fuse with X-type, the other egg fuse with Y-type like shown in this diagram, then 1 male, 1 female twins can be born. Okay? Dizygotic twins are just like siblings. They are born from 2 zygotes, therefore they are called dizygotic twins. They do not look identical, therefore they are called non-identical twins, and they just look as similar as normal siblings, therefore they are called fraternal twins. Okay? Is the dizygotic twins concept clear? Now comes identical twins. Identical twins are also called as monozygotic twins. Here what happens? Only 1 egg cell is released and it fused with only 1 sperm. It will fuse with only 1 sperm, formed only 1 zygote. Therefore, they are called monozygotic. Now the zygote started cell division, formed an embryo. This embryo now splits into 2 embryos. 1 embryo, like 1 bunch of grapes broken into 2 bunches of grapes. Is it clear? You have 2 embryos. The 2 embryos will become 2 babies. The 2 embryos will become 2 babies. Then they are called monozygotic twins because they are born from a single zygote, and they are also called identical twins. Okay? Okay, fine. Guess and tell me, do they look identical? Do identical twins look identical? Everybody? Do identical twins look identical? No. They are only genetically identical. They are only genetically identical. Is that clear? External factors like solar radiations, temperature, humidity, nutrition, all of them can definitely affect. Even their fingerprints can be different. So they are called identical twins because they are genetically identical. They are called monozygotic twins because they are born from the same zygote, And monozygotic twins are always either 2 male or 2 female, or okay, that's all. 2 male or 2, you can never have 1 male 1 female identical twins. Is it clear? Because look at here, if this is XY zygote, you will get 2 XY embryos only. If it is XX zygote, then you will get 2 XX embryos only. So you can never have 1 male 1 female. Identical twins are always either 2 males or 2 females. Is that clear? Now the next topic is cloning. Okay, human cloning. Let us suppose human cloning. Okay, I'll explain human cloning in simple steps. Okay, keep observing the steps. First of all, what do you mean by the word clone? The word clone means copy. We can define it as, clone can be defined as a child of a single genetic parent. Is it clear? Clone can be defined as the child of one genetic parent. Okay? Genetic parent means what? Genetic parent means, okay, the one who gives DNA to the baby. Whereas biological parent is the one who gives birth to the baby, who carries the pregnancy and gives birth to the baby. Is it clear? So, clone can be defined as a child of a single genetic parent. Means what? All 46 chromosomes only from one parent. Is it clear? If you can make a baby taking all 46 chromosomes only from one parent, then it is called as a clone, right? Therefore, we define clone as a child of a single genetic parent. Okay, then now let us see how to do that. Let us suppose you are cloning, okay, a man Mr. X. You are cloning a man called Mr. X or Mr. A. Okay, I am cloning a man called Mr. A. Then step 1, take one egg cell from a donor. You need to take one egg cell from a donor. Tell me, why should I take egg cell from a donor? Because he is Mr. A, he is a male, he cannot release the egg cells. So therefore, take one egg cell from a donor. Step 2. In the step 2, what we do? We remove the nucleus. Remove the egg nucleus. Why to remove egg nucleus? Because if I don't remove the egg nucleus, you will get 23 chromosomes from the donor. But in the cloning, you want to make a baby with all 46 chromosomes only from one person, isn't it? In the cloning, you wanted a baby with all 46 chromosomes only from one person. So if I don't remove the egg nucleus, then I will get 23 chromosomes from the egg donor. Then how can I say that it is a child of a single genetic parent? So remove the egg nucleus. Okay. Now third step. Now what is this? This is egg cell without nucleus, isn't it? Egg cell but no nucleus. Means what? You have only egg cytoplasm. So now tell me, for which purpose we have taken the egg cell from the donor? Did we take the egg cell for the chromosomes or for the egg cytoplasm? We have taken the egg cell from the egg donor for the chromosomes or for the cytoplasm? Yes? We have taken the egg cell for the cytoplasm. Egg cytoplasm is compulsory to make a baby. Egg cytoplasm is compulsory to make a baby. Why? Because egg cytoplasm will have 1,50,000 mitochondria as opposed to our normal cells. It means my skin cell contain only 1500 mitochondria, or a sperm cell contains only 1500 mitochondria. Whereas egg cell contain 1,50,000 mitochondria. Not only that, it will have nutrients, it will have hormones, it will have everything for the fetal development. Egg cytoplasm will have the material needed for the baby to grow. So without egg cytoplasm, you cannot make a baby even artificially. Okay. So step 1, I have taken the egg cell. Why? Because the male do not produce egg cells, so I have taken the egg cell from the donor. Then I removed the egg nucleus. Why? Because I don't want the donor's chromosomes in this egg cell. I want to make a baby with all 46 chromosomes only from this man. Okay. Now you have only cytoplasm. It means you have taken the egg cell for the cytoplasm but not for the chromosomes. Is it clear? Now it is a vacant egg cell. Now to make it a zygote, I want to make this into a zygote. All of you are following? I want to make it into a zygote. So now tell me, what should I add to make this into a zygote? It's a vacant egg cell. Now I want to make it into a zygote. What do I need to make it into a zygote? Yes, clear? No. If you keep egg nucleus, it will become egg cell, na? Anir, if you keep egg nucleus in this, it will become egg cell. But I don't want to make it an egg cell. I want to make it a zygote. To make it a zygote, what should I keep? Everybody. I need 46 chromosomes. Is it clear? Because egg cytoplasm plus 46 chromosomes is equal to zygote. Is that clear? Egg cytoplasm, if I keep sperm nucleus, it will become sperm cell. How can it become a zygote? In the vacant egg cell, if I keep an egg cell, it will become egg cell. In the vacant egg cell, if I keep a sperm nucleus, it will become sperm cell or something else. But I don't want an egg and a sperm. I want to make it into a zygote. A zygote should have how many chromosomes? 46 chromosomes. Do you agree? A zygote should have how many chromosomes? 46 chromosomes. Now tell me, in the Mr. A's body, everybody please, in the Mr. A's body, which cell contain 46 chromosomes, not 23? In the Mr. A's body, which cell contain 46 chromosomes? Right? Cytoplasm is there, na? Donor's cytoplasm is there. We removed only the egg nucleus in the second step. Anir, is it clear? Okay. Yes, Mango, we can do that. So what we have to do is transfer the somatic cell nucleus from the person to be cloned. It means from Mr. A. Is that clear? Okay. Fune entertainment, it is not non-somatic cell. It is a somatic cell. Transfer the somatic cell nucleus into the egg cell. Is that clear? Why somatic cell? Because Mr. A's sperm cell contains only 23 chromosomes, whereas for zygote a zygote needs 46 chromosomes. So which cell in his body contain 46 chromosomes? Somatic cell. Transfer the somatic cell nucleus of the person to be cloned. You want to clone Mr. A? Is that clear? Nothing, I repeat. Okay. Cute girl, I will repeat for you because you are a cute girl. Okay. So first we take one egg cell, right? Whose egg cell you have? I want to clone Mr. A. I want to clone Mr. A. But Mr. A is a male. He will not release the egg cell. So take an egg cell from a donor. Say take my egg cell. Okay. But from that egg cell, remove the egg nucleus, because nucleus contain chromosomes. If you don't remove the egg nucleus, the child will get the features of the egg donor. But clone should be identical to Mr. X. A clone should have all 46 chromosomes only from Mr. X. Is it clear? Okay. The clone should have only one genetic parent, that is Mr. Sorry, if I said X, sorry, Mr. A. Okay. The child should have all 46 chromosomes only from Mr. A. The child should be identical to Mr. A. So I should not keep the donor's egg nucleus. So take her egg cell, remove the egg nucleus. You have egg cytoplasm. It means we have taken the egg cell only for the cytoplasm but not for the chromosomes. Why do you want egg cytoplasm? Because egg cytoplasm is compulsory to make a baby. Why? Because it contains some special features like nutrients, hormones, more mitochondria, etc. Is that clear? Clear till now? You said except 23rd chromosome, all other cells are somatic. Priya, the question itself is wrong. You said 23rd chromosome. That is chromosome. Okay. Every cell contains 23 pairs of chromosomes. 23rd pair is not egg cell. Every cell contains 23 pairs of chromosomes. Is it clear? So Priya, you are getting confused between chromosome and the cell. You are getting confused between what is chromosome and what is the cell. One cell will have 46 chromosomes. Don't confuse between the two. Done? Clear till now? Okay. So all of you tell me what is the step 1? Come on fast. What is the step 1? See, Pavan, I have checked the mic. I have the costliest or the best possible mic. And I am literally tearing my throat and shouting. This is the best I can do. Okay. Try to kindly increase your volume or use a better earphones or something. If I shout little louder, I will have to stop the class after 1 or 2 hours. Okay. Others, let me know, is there any voice issue? Others, let me know, is there any voice issue? Okay, thank you. Thank you so much. Okay. Now, so step 1, take one egg cell. Okay, take one egg cell. Then take one egg cell. From the egg cell, remove the nucleus. Okay. Remove egg nucleus. Keep somatic cell nucleus. Simple 3 steps. Take one egg cell. From that, remove the egg nucleus. Into that, keep the somatic cell nucleus. Why to take egg cell from the donor? Is it clear? Why to take egg cell from the donor? Okay, because you are trying to clone Mr. A. Mr. A is a male. Okay. A male do not release egg cell, but we want egg cell to make a baby. We cannot make a baby without egg cell. So we have taken an egg cell from a donor. Is that clear? Second step, remove the egg nucleus. Why to remove egg nucleus? Because, don't forget this point, clone means copy. The word clone means what? Clone means copy. Or clone means the child of a single genetic parent. The child of a single genetic parent means the child should get all 46 chromosomes only from one person. So if I don't remove the egg nucleus, then the clone will be born with 23 chromosomes from the donor. Is it okay? Not okay. So remove the egg nucleus. Now, it is a vacant egg cell without any nucleus. Is that clear? Clear till now? Okay. Do one thing. Now, I will tell you a small tip to listen my class. A small tip to understand my class. in the first point itself, don't try to understand about what is cloning. That you will understand once I complete all the steps. Okay? At the first step, just make sure that is the first step clear. Then you can understand. Follow the same procedure for all the topics I teach. Okay? At this moment, when I say this, okay, if you try to understand what is clone, you cannot understand. Understand step by step. Once all the steps are completed, you will understand what it is. Is that clear? Okay. Step 1, what is clone, I still did not tell you. So relax, nothing happens. So first clone means what? Copy. Child of single genetic parent. Means should have all 46 chromosomes only from one person. Okay. For that what I am supposed to do? Step 1, take the egg cell from a donor. Why? Because we are trying to clone a man do not release egg cells. Step 2, remove the egg nucleus. Step 2 is remove the egg nucleus. Why should I remove the egg nucleus? Because I want all 46 chromosomes only from this person, Mr. A. Then, now what is this? What you have here? You have egg cell without nucleus. Means you have only egg cytoplasm. Is it clear? So, did we take, for which purpose we have taken the egg cell? For chromosomes or only for the cytoplasm? Yes, all of you? We have taken the egg cell for which purpose? For chromosomes or for the cytoplasm? Only for the cytoplasm. Why egg cytoplasm is so important? Because egg cytoplasm will contain the nutrients and extra number of mitochondria and hormones for the baby's growth. Whatever the baby need to grow, those substances are there in the egg cytoplasm. So we have taken the egg cell only for egg cytoplasm. Now in this, what should I keep to make it into a zygote? It is just a vacant egg cell now. This vacant egg cell plus 46 chromosomes is equal to a zygote. Because a regular zygote will contain what? It will contain egg cytoplasm and the zygote. Is it clear? Right? So, now tell me, if in the Mr. A's body, which cell contain 46 chromosomes, not 23? In Mr. A's body, which cell contain 46 chromosomes? Only the somatic cell. Transfer the somatic cell. Transfer the somatic cell, not the complete cell. Look at here. Transfer the somatic cell nucleus. Cytoplasm is that of the egg cell. Look at here. Egg cytoplasm. Okay, what is there in this white color circle? This is egg cell. What is here? Egg cytoplasm. But nucleus is that of the somatic cell. Whose somatic cell? Mr. A's somatic cell, because you want to clone that person. Now it is a cloned zygote. Then you may get one doubt. Ma'am, in that case, is it a baby made without the sperm? Yes, here no sperm used. Without sperm. Then you may ask me, ma'am, can we make a baby without the sperm? No, not naturally. Naturally, we cannot make a baby without the sperm, but it is a technique. It is artificial. It is artificial. It is a technique that we do in the laboratory. So, in the laboratory, we can do that. Okay, Kali Charan, kaise hoga bhaiya? Hoga bhaiya, no problem. Okay, because nature में नहीं होगा, पर laboratory में होगा, हम कर सकते. We can do it in the laboratory. Okay, we can keep the somatic cell nucleus in the, we have micro injections. Okay, collect the somatic cell nucleus, inject it into this egg cell cytoplasm. Now artificially, it is not a natural zygote, but it is a cloned zygote. Is that clear for all of you? Yes? Then clear? Right. Okay. So this is called as a cloned zygote. Then it is artificial only till you make a zygote. You cannot make a baby in the laboratory. In the cloning we don't make a baby in the laboratory. It is only till the zygote. Then this zygote is implanted into the womb of some lady. Then after 9 months the baby will be born. Okay, is that clear? That's all. That is the procedure. Okay. Now I will ask you a few questions. Let us suppose you have taken, I'll ask you a few questions. Okay, you have taken Mr. A, given the somatic cell nucleus. Mr. A has given the somatic cell nucleus. Then, okay, Miss B has given the egg cytoplasm. Okay, Miss C, okay, carried the pregnancy. And now the child is the clone of. Tell me, child is the clone of? Very good. The child is the clone of Mr. A. Okay, because Mr. A has given somatic cell nucleus, means 46 chromosomes. Therefore, he is the genetic parent. Okay, that's all. That is the procedure. Is it clear? Okay. Now, this cloning will be, okay, now let us compare between a natural zygote and a cloned zygote. Is it clear? Let us compare between a natural zygote and a cloned zygote. Okay. Do you agree both of them contain natural zygote, cloned zygote? Both of them contain egg cytoplasm plus 46 chromosomes. Do you agree that is a common point? Clear? We need to pick up the speed, otherwise I will not be able to complete all the planned topics. Okay. If you keep saying repeat, I can explain it, no problem, but I cannot complete the topics. Only cloning I will explain you clearly, we will end up doing that. So you should also keep an extra effort to increase your focus levels, be more alert, okay. Keep following the instructions and all that. Is it clear? Okay. Now, so normal zygote, cloned zygote, both will have egg cytoplasm plus 46 chromosomes, right? Okay, right. Then, but what is the difference? In the egg cell, you have, in the normal zygote, you have the 46 chromosomes from egg nucleus and sperm nucleus from two people in the normal zygote. But in the cloned zygote, you are getting it from the somatic cell nucleus, from a single person. That is called as cloning. Is that clear? Then, cloning can be of two types. Okay. Cloning can be of two types. One is reproductive cloning. The other one is, okay, look at here. Okay. Fine. So, this is cloned zygote. Now, if the cloned zygote is implanted into the womb to get a cloned baby, that is called reproductive cloning. That is called reproductive cloning. Why that is called reproductive cloning? Because you are reproducing a baby. You are making a baby. But if the cloned zygote grows into cloned embryo in the laboratory, okay, and use them to make organs for transplantation, use it to make organs for transplantation. That is called as therapeutic cloning. That is called therapeutic cloning. Means cloning for therapy. Which therapy? Organ transplantation is a therapy. For example, a person's kidney failed. If a person's kidney failed, what is therapy there? What is treatment there? The treatment is organ transplantation, kidney transplantation. Using this cloned embryo, if I make a new kidney for transplantation, that is called as therapeutic cloning. So, once you got the cloned zygote, okay, grow it into embryo, make organs for transplantation, therapeutic cloning, or implant it into the womb and get a baby, that is called reproductive cloning. Is it clear? Right? Then, reproductive cloning of the human beings is not permitted. Okay, human cloning is not permitted, remember that point. On 8th March 2005, the United Nations General Assembly adopted a declaration on the human cloning which states that all forms of human cloning are incompatible with the human dignity, okay, with the protection of the human life. So therapeutic cloning, reproductive cloning, both clonings are banned. Human cloning is not permitted. Then why are we discussing this? Because animal cloning, same procedure, animal cloning is possible. For example, American scientists in 2020 announced the first cloning of an endangered animal, a species which is about to become extinct, endangered animal, okay, called black-footed ferret, that was created using the egg cells which are stored, from the cells which are stored 30 years ago. How they are stored for so long? Okay. How they are stored so long? In liquid nitrogen at minus 196 degrees Celsius. Is it clear? Clear till now? So I repeat, there is one animal called black-footed ferret. It is an endangered animal. They have collected the egg cells and somatic cells of that species and preserved in the liquid nitrogen. 30 years later, using cloning, they created a black-footed ferret organism. Like this, we can use cloning to revive an extinct species or create the endangered species. This is one application of the cloning, but human cloning for all purposes is banned. Clear till now? Right. Moving on to the next topic. This is 3-parent baby. Cloning is a single parent baby. Now this is a 3-parent baby. Let us see who are those 3 parents. Okay. To explain this, I will give you some more basics. Okay. First we need to understand what is a gene. Okay. What is a gene? Gene is any functional sequence of nucleotides on the DNA. Is that clear? It means you have if this completely is DNA, then one small part of it is called as gene. So what is it? For example, this is producing insulin. It is producing insulin. Okay. Means, is it useful or useless sequence? I said it is producing the insulin. So is it a useful sequence or a useless sequence? Useful sequence. So therefore, any functional sequence of nucleotides, any functional sequence of nucleotides on the DNA is called as a gene. Is it clear? We have genes in the DNA just like we have words in a sentence. Just like we have words in a sentence, we have genes in the DNA. Okay. Whereas let us suppose the next sequence is not coding for anything. It is not producing anything. Not producing a protein. not producing a protein, then it is called as junk DNA. We don't call it as a G. Is that clear? Then it is called junk DNA. It is not called as a gene. Clear till now? Right. Then what is a mutation? Mutation is a change in the gene sequence. Is it clear? What is a mutation? If I compare a gene with a word, then mutation is like a spelling mistake. Mutation is like a spelling mistake. Means it is a change in the gene sequence. For example, this should be the sequence. Let us suppose this is the correct sequence. Okay. Then it should be TACGGAT. Why? Because I already told you about the base pairing rules. The base pairing rules are A will pair with T. Okay. Then T with A, G with C, C with G, like that. Okay. This is the correct sequence. But now let us suppose, okay. Now let us suppose that there is one wrong letter here, G. That is called as a mutation. Is it clear? Change in the gene sequence. What happens? It will alter the gene function. Means the gene will fail to do the function that it is supposed to do. A gene fails to do the function. Okay. Then in our cells, most of the DNA is present inside the nucleus. Tell me, where is most of our DNA present? Most of our DNA is present inside the nucleus, right? But a little amount of DNA, 0.1% DNA is also present in the mitochondria. Is it clear? Little amount of DNA is also present in the mitochondria. Now, answer these questions. Can the mitochondrial DNA get mutated? Can we get a mutation in the mitochondrial genes? Yes. The mutations in the mitochondrial DNA cause these diseases: Alport syndrome, Leigh syndrome, etc. Okay. The mutations in the mitochondrial DNA cause Alport syndrome, Leigh syndrome, etc. Therefore, these are called mitochondrial diseases. Clear? They are called mitochondrial diseases. Done? Now, I am explaining the next point. Once again, I am telling you, keep collecting the points. Once all the points are completed, you will understand what it is. At the first step, you will not understand what is three-parent baby. Okay? So, to understand three-parent baby, these are all the points that you need. You are stitching them together. Okay? What is a gene? What is a mutation? We have little amount of DNA in the mitochondria. If the mitochondrial DNA gets mutated, we get these diseases. These are some parts of the information. Then next point. Is it clear? Next point. Okay. In general, what is fertilization? Fertilization means fusion of the egg cell with the sperm cell, isn't it? This is egg cell. This is sperm cell. These two will fuse to form a zygote. That is called fertilization, but that is not completely correct. Okay. During fertilization, what happens is, look at here, this is egg cell with 23 chromosomes. Okay. Now look at here, what is this? This is sperm cell. Again with 23 chromosomes. We generally think that egg and sperm will fuse like this, but they don't fuse like that. Only the sperm nucleus goes into the egg cell. Is it clear? Now the egg cell became a zygote. The egg cell became a zygote. Okay. So now tell me, is the sperm cytoplasm present in the zygote? Do we have the sperm cytoplasm in the zygote? Yes, everybody? Do we have sperm cytoplasm in the zygote? No. Okay. the total cytoplasm of the zygote is only from, the total cytoplasm of the zygote is only from the egg cell. Do you agree? It means only nucleus of the sperm goes into the egg cell. Is it clear? And another small request, don't try to take the running notes because it is a marathon session. I have to proceed extremely fast. So in this, if you try to write down the points, you cannot focus on the class. We will end up in unnecessary repetitions. All the points that you want is there in the notes. You can download the PDF. It is not possible to write down all the points in this class. So kindly complete focus on the class, please. Okay. So only the nucleus of the sperm cell goes into the egg cell, isn't it? So now, okay, where are mitochondria located? All of you, please. Mitochondria are in the, yes, mitochondria are in the cytoplasm. Therefore, a child gets all mitochondria only from, yes everybody, mitochondria are in the cytoplasm. Sperm cytoplasm is not going into the egg cell. The zygote contain only the egg cytoplasm. Okay, so a child will get all his mitochondria only from the, okay, only from the mother. A child get all the mitochondria only from the mother. Is that clear? Okay. So it means, can a father inherit mitochondrial diseases? Here, remember the previous point I said. Alport syndrome, Leigh syndrome we get because of the mutations in the mitochondrial DNA. Can a father inherit mitochondrial DNA to the children? No. Okay. So, mitochondrial diseases. Mitochondrial diseases follow maternal inheritance. Maternal inheritance, it means only inherited only from the mother, not from the father. Is it clear? Then, now I have one question here. Okay, I'll tell you a situation, tell me a solution for this problem. There is a woman, now she wants to have a baby. Okay, she wants the baby to look like her, but should not get her mitochondrial disease. Is it clear? There is a woman, she has a mitochondrial disease. Now she wants to have a baby. She wants the baby to look like her, features should be that of her, but should not get her mitochondrial disease. For this, I have to take something from a donor. Can you tell me what should I take from the donor? Yes? For this, if this lady wants the baby to have her features, but not her mitochondrial disease, then what should we take from a donor? Egg cytoplasm. But taking egg cytoplasm is difficult. So what you do is take the egg cell from the donor and then remove the egg nucleus. Why? Because this lady wants the baby to have her features. She don't want the baby to have the features of the donor. Only mitochondrial disease she want to avoid. So remove the egg nucleus. It means we took the egg cytoplasm having 1,50,000 mitochondria. Is it clear? Some people ask me, ma'am, why don't you just replace the mitochondria? Why to take the complete cytoplasm? Not possible, because you have 1,50,000 mitochondria, means you have to 1, 2, 3, 4 karke, you have to add 1,50,000 mitochondria. It is like showing, if somebody asks where is your nose, I should show like this, I should not try to show like this, it will be like that. Is it clear? So therefore simplest procedure is take the egg cell, remove the egg cell from a donor, remove the egg nucleus. Means you have taken 1,50,000 mitochondria and then you keep, okay, the sperm nucleus from the father. Sperm nucleus from the father and then egg nucleus from the mother. Look at here, mitochondrial DNA is from the donor, 23 chromosomes of egg nucleus from the mother, 23 chromosomes of sperm nucleus from the father. So how many genetic parents are there in this case? How many people are contributing the genetic material to this baby? No, it is not cloning at all. Perfect, it's not cloning at all. Because cloning one genetic parent, okay, only one genetic parent. Here you have 3 genetic parents here. Who are the 3 genetic parents? Perfectly, is that clear? Okay, who are the 3 genetic parents? Who are the 3 genetic parents here? The first genetic parent is mitochondrial DNA from the egg donor. Mitochondrial DNA from the egg donor. Ashwini, like how mitochondria is inserted? We have micro injections, beta, we can inject it. But why do we need it? We don't have laboratory for UPSC exam. We don't have laboratory work. So focus on the necessary points, please. And I really can't increase my voice. I'm really shouting. So if the voice is not audible, we have only one solution, stopping the class. Can we, or should we continue? If the voice is really not audible, okay, let us, okay, because it's beyond my capacity, I can't shout more, okay. Right, so first genetic parent is mitochondrial DNA donor, okay, the one who has given the egg cytoplasm, because that is also, who is a genetic parent? The one who gives DNA. So the egg donor is the first genetic parent, and then, okay, the one egg cytoplasm donor is the first genetic parent. Mother is the second genetic parent because you get 23 chromosomes from the mother. Father is the third genetic parent because you will get other 23 chromosomes from the father. There are 3 genetic parents here, so it is not at all similar to cloning. Then what is the purpose of this? Okay, so therefore we call it as 3-parent baby. Why we do the 3-parent baby technique? This is only to avoid mitochondrial diseases. This is only to avoid the mitochondrial diseases. Is it clear? And this is also called as mitochondrial replacement therapy. It is also called mitochondrial replacement therapy because we are replacing the mother's defective mitochondria with the healthy mitochondria of the donor. It is called mitochondrial replacement therapy because we are replacing the mother's defective mitochondria with the healthy mitochondria of the donor. Okay? Therefore, it is also called mitochondrial replacement therapy. Clear till now? Yes. The other name is pronuclear transfer. This is also called as pronuclear transfer. Why this is called pronuclear transfer? Because pronuclei means the egg and sperm nuclei, okay, which are still fusing, means it's during the fusion, okay. It means they are not like this, 23, 23, they are not like this. They are not like this, 23, 23, not completely fused, not unfused, but they are like this, partly fused, still fusing, 23 here, 20, this is egg nucleus, this is sperm nucleus. Is it clear? Clear till now? The egg and sperm nuclei that are still fusing is called as a pronuclei. This pronuclei will be transferred. Okay, therefore it is called pronuclear transfer. We replace mother's defective mitochondria with the healthy mitochondria of the donor, therefore it is also called mitochondrial replacement therapy. This zygote contains the genetic material from 3 people, therefore it is also called 3-parent baby. The purpose is to avoid the inheritance of the mitochondrial diseases. Now tell me, if this zygote is implanted into the womb of a 4th lady, then can we call it as 4-parent baby? Is it clear? If this zygote is implanted into the womb of a 4th lady, some 4th person is involved, can I call it as a 4-parent baby? No, we cannot call it as a 4-parent baby, because if I implant this zygote into the womb of a lady, that lady will be called as biological parent, not genetic parent. Biological parent is the one who gives birth to the baby, whereas genetic parent is the one who gives genetic material to the baby. Genetic material is only coming from 3 people, so whoever carries pregnancy, that person need not be counted. We call it as 3-parent baby. So who carries and gives birth to this baby is immaterial. Any lady can carry and give birth to this baby. She is biological parent. Clear till now? Okay. Then now, we have a few previous year questions based on this topic. In the context of hereditary diseases, consider the following statements. Passing on mitochondrial diseases from the parent to child can be prevented by mitochondrial replacement therapy either before or after fertilization? Yes, correct. Okay, means what? We can do it in 2 different ways. It means this is donor's egg cell. Then what you do is, okay, this is fertilized. Okay, this is fertilized, already fertilized. It means already fused. Egg plus sperm nuclei. This is egg nucleus. This is sperm nucleus. Already fertilized, already fused. That you transfer is one process. Okay. Second process is, you take the donor's egg cell. Then you first transfer the egg nucleus. Then you transfer the sperm nucleus. Okay. Then they fuse, they fertilize after the transfer. Is it clear? Either you fertilize them outside and together send this. Is it clear? Fertilized egg and sperm. First fertilize it outside and then send. Or you send into the donor's egg cell, you insert them into the donor's egg cell and they will fertilize after you insert it into the donor's egg cell. Okay, both are possible. Is that clear? So there are two ways of doing it. First fertilize it and then inject, or first inject and then they will fertilize inside the egg cell. This is first statement correct. A child inherits mitochondrial diseases entirely from the mother and not from the father. I already told you mitochondrial diseases follow maternal inheritance, because during fertilization the sperm cytoplasm is not coming into the zygote, only the sperm nucleus goes into the zygote. Mitochondria of only the mother will go into the baby. Clear? So both 1 and 2 are the correct answers. And then yes, I have given the answer already highlighted, because the target of this phase of Conquer Prelims is not to make you practice the previous year questions. We already completed the phase 2 on Unacademy platform as a free initiative special classes, where in each class I discussed 30 multiple choice questions, 20 to 30 multiple choice questions, where I have given all the questions for the practice. I did not highlight the answer in that. Here the target is only the revision. So just to show you that this type of questions are coming. Is it clear? Now, in the context of recent advances in the human reproductive technology, pronuclear technology is used for prevention of mitochondrial diseases in the offspring. Done? That's all. With that we completed the 3-parent baby. Now moving on to the next topic. The next topic is assisted reproductive techniques, or also called as artificial fertility techniques. Is it clear? Assisted reproductive techniques or artificial fertility techniques. Let us suppose in a couple the wife is unable to release the egg cell. In a couple the wife is unable to release the egg cell, or the husband is unable to release the sperm cell. Naturally they are unable to have, it's not possible for them to have a child. So do we have any medical procedures to help them have a child? Yes, those are called artificial fertility techniques or assisted reproductive techniques. Can we proceed? Clear till now? Okay. In the assisted reproductive techniques, the first one is called as cell banks. Cell bank. Okay. What is a cell bank? Just like in an eye bank we preserve the eyes, same way here in the cell bank we preserve the cells. Cells are preserved in the liquid nitrogen tanks at a temperature of -190 degrees Celsius. Is it clear? Eggs and sperms are preserved in the liquid nitrogen at a temperature of -196 degrees Celsius. Here we can even preserve them safely for up to 100 years also. Up to 100 years you can preserve them. Clear till now? Then, now what we will do using that preserved egg cells? The first one is called as in vitro fertilization. The word in vitro means in the laboratory. In vitro means in the laboratory. So here what they do is, okay, I have shown you the procedure here, go through this. Okay, check the first step first. Step 1. The woman is given hormone treatment. Why? Because I told you in most of the cases a woman releases only one egg cell. But if you start it with only one egg cell, the success rate is very less. So we better start with more egg cells. So for that what we do is we give a hormone injection. With that hormone injection what happens is more egg cells will be released. Is it clear? More egg cells will be released. Then step 2, multiple eggs are taken from the woman's ovaries. Okay, multiple egg cells are taken from the woman's ovaries. You are able to hear this, please? What is this structure? These are ovaries. Then what is this tube? These tubes are called fallopian tubes. The tubes are called fallopian tubes. What is this structure? This structure, this part is called uterus. That is called uterus. Okay, this is called cervix. This is called cervix. Okay, cervix means opening of the uterus. What is step 1? Step 1 is give hormone injections to a woman. Now the woman release more egg cells from the ovaries instead of a single egg cell. Step 2, multiple egg cells are taken from the woman's ovaries. Step 3, in the laboratory, the eggs are mixed with the sperms in a petri dish, in a culture dish or petri dish or a test tube like this. Is it clear? These multiple eggs are fertilized with this multiple sperms in this culture dish. That is step 3. Okay? Right. Then what is step 4? In the step 4, the zygotes are formed. Zygote is formed like this. Okay? Here you have taken the egg cell from the wife. But her husband is infertile. So you have taken the sperm cell from a donor. Sperm cell from a donor. Is that clear? Then this formed a zygote. This formed a zygote. This zygote is now incubated. Incubated. Okay, it is kept in the incubator if you observe here. It's an incubator. Why? To bring this to the womb temperature. To bring this to the womb temperature. Then after incubation, what you can do? You can do two things with this. First, okay, you can implant it. Implant it into the womb. Or second thing, you can freeze it. You can freeze it. How long? Recently, a baby was born from an embryo which is frozen 30 years ago. Okay, freeze for decades if you want. And then implant it. And then implant it into the womb. Then it is called FET. FET means frozen embryo transfer. Is the procedure clear? FET means frozen embryo transfer. Okay. So once embryo is ready, either implant it into the womb directly or freeze it in the liquid nitrogen and implant it whenever you want. So that procedure is called in vitro fertilization. If you do this at the zygote level, you can call it as IVF. Rather, if you grow this into an embryo in the laboratory itself, if you grow this into the embryo in the laboratory itself, then it is called embryo transfer technique. So between IVF and embryo transfer technique, there is only one difference. At the zygote level, if you transfer, it is called IVF. At the embryo level, it means after some 1 or 2 days, once embryo is formed, and then if you implant it, it is called as embryo transfer technique. Okay, are both the techniques clear? In vitro fertilization and embryo transfer, both are clear. Okay. Then next one is called as artificial insemination. Artificial insemination. In this case, let us assume that a man is able to release the sperm cells. His wife is able to release the egg cell. No need of an egg donor or a sperm donor. The only problem is maybe the man is suffering from a condition called as oligospermia. Oligospermia means what? Lesser number of the sperms. Oligospermia means lesser number of the sperms. Then what happens? The pregnancy can get delayed. Pregnancy delayed. Then now what we do? We collect the sperm. We collect the sperm artificially and inject it inject it into, okay, different places you can inject. Look at here. Try to answer this. If I inject the sperm into the cervix, then what we can call it as? If I inject it into the cervix, here if I inject, that is called ICI. Intracervical insemination. Insemination means injecting the semen into the cervix. Is that clear? If I inject it into the uterus, that is called IUI. Is it clear? That is called IUI. Intrauterine insemination. Is that clear? Okay. Then like this. Intracervical insemination is injecting the semen into the cervix. Intrauterine insemination is injecting the semen into the uterus. Okay. If you inject it into the gametes, if you inject this into the fallopian tube, here if you inject, that is called gift. Gift means gamete. Gametes means what? Okay, note down. The word gametes means female gametes are nothing but the eggs. Male gametes are nothing but the sperms. Female gametes are eggs and male gametes are sperms. Okay. So, we are injecting the sperm into the fallopian tube. Therefore, it is called gamete. Okay. Observe it. Gamete means sperm. Intrafallopian. Intra means inside. Intrafallopian transfer. Gamete intrafallopian transfer. Okay. Sperms and eggs are mixed and injected into the fallopian tubes. In all the three cases, we can call it as artificial insemination. Okay. Because of oligospermia, lesser number of the sperms, pregnancy is getting delayed. Now the couple want the baby soon. So I will artificially collect the semen and inject it into the woman's body. Depending on where do we inject, we have intracervical insemination, intrauterine insemination, or gamete intrafallopian transfer. Is that clear? Then next one is called surrogacy. Okay. In surrogacy, what happens is, for example, like if let us suppose we have a famous case like Priyanka Chopra, Nick Jonas, and they had a baby through surrogate mother. It means what? The egg and sperm are that of this couple, but the baby is carried by some other lady. Now tell me, can that lady keep this couple's baby forever, or she is giving birth to the baby for this couple? Is it clear? Chirag, we are doing insemination only in the gift also. In the gift also, sperms are injected. Eggs and sperms are mixed and injected. Okay? Yes, what was the question? So, is that lady, did that lady give birth to the baby for this couple or to keep for herself? Yes, everybody? So, surrogacy is any case. If I will give birth to a baby for some other couple, that is called surrogacy. Okay? If any lady gives birth to the baby for some other couple, not for herself, she is just renting her womb. She is just lending her womb. That is called surrogacy. She is called either surrogate mother or gestational carrier. Is that clear? She is called surrogate mother or gestational carrier. Okay. And recently in the year 2021, the Indian government passed two different acts. One is Assisted Reproductive Technologies Act and second one is Surrogacy Act in the year 2021. Okay. So, this is some factual content, you can go through this. Is it clear? Okay. So, you have to read about these two acts, Surrogacy Act and Assisted Reproductive Technologies Act. Both are passed on the same day. What is the difference? ART Act covers all, that is in vitro fertilization, okay, or frozen embryo transfer, or gift, or ICI, or IUI, or surrogacy. All assisted reproductive technologies are covered in the ART Act. But Surrogacy Act is exclusively for surrogacy. Is it clear? What is okay, now let us proceed to the next topic, that is artificial wombs. Can we proceed? Artificial womb. Artificial womb is like this. Artificial womb can also be called as a biobag. Yes, tell me, the artificial womb is also called as? Artificial womb is also called as a biobag. So what is it? It looks like this. Like say for example, this is the biobag. Inside the biobag you can see some liquid. What is this liquid here? The liquid is nothing but amniotic fluid. It means in this particular bag, we try to create exactly the womb-like situations. Is it clear? Inside the biobag, we want to create the womb-like situations. What will be there in a natural womb? Amniotic fluid will be there. So we keep the amniotic fluid here. Okay. The baby's lungs are not still working. So we use an external oxygenator. Look at here. This is an oxygenator which will add oxygen to the blood. It means this machine will work like the lungs. Is it clear? This machine works like, yes, tell me. This machine works like lungs. Then into the blood, using a drip bag, we are adding nutrients into this blood. Did you notice? We are adding nutrients into this blood because this baby's digestive system is not yet developed. Okay, this fetus cannot eat food and digest. Okay, so therefore directly the nutrients are added into the blood. And then waste material is removed by dialysis. Is it clear? The toxins accumulated as the day-to-day metabolism is going on, lot of toxic material will be accumulated in the body of this fetus. So from the fetus we have to remove this toxic material, okay, waste accumulated through a process called dialysis. Is it clear? So it means we take a bag-like structure, in that we keep amniotic fluid, we maintain the temperature, humidity, pressure and all these aspects exactly like that of the woman, like that of the womb. And because the baby's lungs are not still working, we use a machine called oxygenator to add blood to that baby. And we also add nutrients into the same blood because the baby cannot even digest the food. And the baby cannot even excrete the waste material, therefore we remove the waste from the baby's body through the dialysis procedure. It means we artificially do everything that naturally occur in the womb in a natural pregnancy. Is it clear? In a natural pregnancy, what all occurs in a natural womb, here we try to do them artificially. This is called as an artificial womb. Do we already have artificial wombs? Yes. But is there any human baby born in an artificial womb? No. Is there any fetus developed in the artificial womb? Yes. A lamb fetus was developed in the artificial womb. Is it clear? As of now, only a lamb fetus was developed in the artificial womb. Why they tried with lamb? Because it is a very common organism. If this is successful, actually they want to try it on the endangered species or the extinct animals to grow their babies. Is it clear? Yes, clear till now? Then, okay. Now, artificial fertility techniques in biodiversity. How can we use artificial fertility techniques in the biodiversity conservation? Okay. Did you hear about this northern white rhino? Okay, I'll explain it. Listen carefully. There is one species called as white rhino. There is one species called white rhino. What is this? This is a species. In this there are two subspecies. In this there are two subspecies. One is called southern white rhino. The other one is called as northern white rhino. Southern white rhino, northern white rhino. Northern white rhino is extinct in wild. Means in the nature it is extinct. Artificially you can save it, but using natural procedures you cannot save it. Because in this, the last male already died. You have two females but they became old and they cannot reproduce. Is it clear? There are two females but they became old. Okay. So they cannot reproduce. So what they have done is, they used southern white rhino, SWR, as a surrogate to give birth to the northern white rhino. But this is not yet successful. Is it clear? The effort is not yet successful. Is it clear? There is a species called white rhino. In that you have southern white rhino, northern white rhino. The northern white rhino is about to become extinct, and in the nature it already became extinct because the last male died and you have two females, but they also became very old, not in the reproductive age. So they have collected the egg and sperm from northern white rhino, means like this. Look at here. Egg from the northern white rhino plus sperm from the northern white rhino. Then it formed a northern white rhino zygote. But this zygote is transferred into the womb of the southern white rhino. Is it possible? Possible but difficult and not yet successfully done. They are trying to do that. Is that clear? Then, so this is how we can use artificial fertility techniques in conserving the biodiversity. Okay, we can use artificial fertility techniques in conserving the biodiversity. Okay, can we move on to the next topic? The next topic we are discussing is stem cell technology. Okay. The next topic is stem cell technology. Then what are the stem cells? Okay, first we have to understand what are the stem cells. Stem cells are the cells that can be converted into other cell types. Is it clear? Stem cells are the cells that can be converted into the other cell types. Perfectly okay. I'll give you the break after the stem cell technology. Okay? After the stem cell technology, we can take the break. Cells that can be converted into the other cell types. See, just to motivate you, okay, see, you are comfortably sitting in a place and watching the class. Understand about my situation also. On the one side, I have a conscious that some people are saying the mic is not working properly. Half of my mind is on the mic. Half of my mind is on the topic. Okay, and shouting to my fullest energy. So try to take little motivation. Okay, and try to completely focus on the class and let us try to make it maximum productive possible. Is it clear? So, stay motivated. I know it is difficult to concentrate in the lengthy classes, but you can do it. Is it clear? Right. So, stem cells are the cells that can be converted into the other cell types. It means, for example, imagine these are the stem cells. You can convert a stem cell into a liver cell, or you can convert a stem cell into a pancreatic cell, or you can convert a stem cell into a heart cell, or you can convert the stem cell into a skin cell. So, stem cells are those cells which can be converted into the other cell types. Is it clear? It means these are like raw material. These are like raw material from which we can make organs. Is it clear? These are the raw material from which we can make the organs. Okay. The class will have a minimum duration of 6 to 7 hours. Maybe it will extend till 1 o'clock. Minimum till 1 o'clock it will extend. Okay. So, the stem cells are the raw material. Clear till now? Okay. Now, what are the stem cells? They are undifferentiated cells or partially differentiated cells. They are undifferentiated or partially differentiated cells. It means they are nothing. They are nothing, like a white wall. So, is it easy to paint a red wall into green or a white wall into green? Is it easy to make a white wall into green or a red wall into green? It is easy to make a white wall into a green. Why? Because white wall is nothing. So, it can be converted into anything. Just like stem cells are nothing, they are not differentiated yet, they are undifferentiated. So, they can be differentiated into any cell you want. Okay? It's like a white wall, you can paint any color on that. That is the meaning of the word stem cells. Is it clear? Then, depending on how many other cells they can get converted into, the stem cells can be divided into different types. First type are called totipotent precursor stem cells. The name sounds very lengthy and difficult, but just look at here. Totipotent precursor stem cell. Toti means total. Potency means capacity. Precursor means raw material. Cells means they have complete capacity. Means these are the cells that can get converted into all the cell types. Means they can even make a complete organism also. The only example we have is zygote. Is it clear? The only example we have is zygote. Zygote is totipotent. From the zygote you can make any cell in the body. You can make a complete organism also. You can make a complete baby also. Is the term clear? Then next one is called pluripotent precursor stem cell. Okay. So, easy to remember also this word potent precursor stem cell repeated. Just in the place of toti you have to keep pluri. Pluri means what? Plural. Singular means one, plural means many. These are the cells that can get converted into most of the other cell types. Not all, but most of the cells you can make. Means, for example, you may not be able to make brain cells, but you can make heart cells, liver cells, kidney cells, pancreatic cells. All the cells you can make except a few. Okay. Example for such pluripotent precursor stem cells are embryonic cells. Look at here. In the totipotent, all is the keyword. In the pluripotent, most of the is the keyword. Next, in the multipotent, okay, many is the keyword. It means cells that can be converted into many other cell types. It means here 100%, here 99%, okay, here only some 90% of the other cells you can make. Is that clear? Yes, Aman, we can make them. Clear till now? From a cell, if you can make 100% all the cells in our body, if you want, you can even make a complete organism. That is called totipotent. From some cells, if you can make most of the cells in our body, but not all, then it is called pluripotent. Okay, from the cells, if you can make many cells in our body, but not a good number of them you can make, then they are called multipotent precursor stem cells. Okay, then oligo means few. I already told you. In which context I told you? Oligospermia I said. It means lesser number of the sperms. Now oligopotent means what? Cells that can be differentiated into only few other cell types are called as oligopotent precursor stem cells. Example, bone marrow cells. They can make only few cells, right? So they cannot be used to produce the organs. You cannot make a liver, kidney, heart, pancreas from the bone marrow cells. Then for which purpose we use them? They can be only converted into different types of blood cells. Therefore, they can be used in the treatment of the blood cancer. We use the bone marrow stem cells for the blood cancer treatment. Is it clear? Okay. Then next you have induced pluripotent precursor stem cells. Okay, fine. Tell me, what is the example for totipotent? Yes, all of you. What is the example for totipotent? Zygote. What is the example for pluripotent? Embryo. Example for multipotent? Umbilical cord. What is umbilical cord? Imagine this is the uterus and imagine this is the fetus, okay, and then this is the placenta. From the placenta to the fetus there is a tube. That tube is called as umbilical cord. That tube is called as umbilical cord. Is it clear? Right? Umbilical cord is a connection between the mother and the baby. Mother provides nutrition to the baby through this umbilical cord. When a baby is born, this umbilical cord will be cut. Then the blood comes. That blood is preserved in the liquid nitrogen. Is it clear? Umbilical cord blood cells are stored in the liquid nitrogen and can be used to make organs. Is that clear? On the whole, what is the target of the stem cell technology? Okay, the target of stem cell technology is the target of the stem cell technology is to make copies of one's own organs for transplantation. Is it clear? For example, I want a kidney. Okay, kidney failed. I want a kidney. Okay, I cannot take a kidney from another donor because the donor's kidney will not perfectly match with my body. Sometimes we may get a suitable donor, but except if you have an identical twin, there is nobody whose kidney will perfectly match with your body. In that case, how it will be if I can make another copy of my own kidney? if I can make another copy of my own kidney. That is the target of the stem cell technology. If you have zygote, you can make all organs like that. If you have embryo, you can make most of the organs like that. If you have umbilical cord blood cells, you can make many of the organs like that. If you have oligopotent bone marrow cells, you can make only few other cell types but not organs. You can make different types of the blood cells which can be used in the blood cancer treatment. Is that clear? Okay. Just keep responding please. Is that clear? Then first tell me, is it legally permitted to collect the zygote like that? Can I collect the zygote and preserve it in the liquid nitrogen? Not permitted. Can I collect embryo and preserve it in the liquid nitrogen? Embryo from the womb and preserve it in the liquid nitrogen for making organs in the future? Not permitted. Is it clear? Can we preserve umbilical cord stem cells in the liquid nitrogen? Yes, it is legally permitted. If I want to preserve the umbilical cord stem cells, when should I preserve them? At the time of the birth. And till when they can be preserved? Up to 100 years safely you can preserve it. Okay. For example, when this baby is born, complete focus here. Okay. When this baby is born, I preserved the umbilical cord cells. As long as this baby is alive, in this baby's life, any organ you want, you can manufacture from these stem cells and surgically arrange in that baby's body. That is the advantage of the stem cell technology. Is it clear? Right? For umbilical cord stem cells, there is no need of any permission because umbilical cord is something that we will otherwise throw. Simple, baby born, umbilical cord cut, put the umbilical cord blood cells in the liquid nitrogen, preserve it for 100 years. In this baby's lifetime, if you want any organ, you can manufacture them using those cells and can surgically arrange that organ into this baby's body. Is it clear? Clear till now? Then, so now tell me, do we have, yes, now tell me, do I have my zygote today? No. Okay, my zygote became me. Do you have your embryo today? No. Your embryo became you. Do you have your umbilical cord today? No, because your umbilical cord was thrown when you were born. Do you have your bone marrow today? Yes, but it cannot be used to make organs. So if today you or me need an organ, then what to do? Then the solution is called as induced pluripotent precursor stem cells. Is it clear? Induced pluripotent precursor stem cell. Okay, right? What is this induced pluripotent precursor stem cell? Okay, answer this question. If the embryo gets converted into organs or somatic cells, what do you call that process as? If the embryo gets converted into somatic cells, what is that process called as? That process is called as differentiation. Then, what will you call? Just try to give it a name. What will you call if the somatic cells convert back to embryo? Then they are called as? If the somatic cells convert back to embryo, then they are called as? That process is called as de-differentiation. We call that process as de-differentiation. Is that clear? Reverse process. So, what you do is, you can take a somatic cell. I will explain this in 3 simple steps. Step 1, take a somatic cell. Is the first step clear for all of you? Then step 2, okay, what we do is inject embryonic transcription factors. Embryonic transcription factors into the somatic cell. Is it clear? Then what happens? The embryonic genes starts express means what? They undergo transcription. They undergo transcription, translation. I mean, they produce proteins. Embryonic genes will start producing proteins. Where? In the somatic cell. Is that clear? Remember the process. First take the somatic cell. Into that inject the, means like you have taken the somatic cell here. We have taken the somatic cell here. Into the somatic cell you are injecting the embryonic transcription factors. What they do? They will help the embryonic genes undergo. Okay, look at here. With the help of this, the embryonic genes will start expressing. The embryonic genes will produce proteins in the somatic cell. Is it clear? Once the embryonic genes produce proteins in the somatic cell, now the somatic cell behaves like, or now the somatic cell is like, now the somatic cell is like the embryonic cell. Is that clear? Take the somatic cell, inject embryonic genes transcription factors into it. Then in the somatic cell, the embryonic genes will start expressing. The embryonic genes will start producing the proteins. Now because embryonic proteins are produced in the somatic cell, now the somatic cell is like embryonic cell. Is it clear? Means the somatic cell is like pluripotent cell, isn't it? Because embryonic cells are pluripotent cells. Got it? Now the somatic cell became like pluripotent cell. Is it clear? Embryonic transcription factors can be synthetically manufactured, Avinash. Okay? Take the somatic cell, inject embryonic gene transcription factors into that. Then embryonic genes will start producing the proteins. In the somatic cell, embryonic proteins are produced. So now the somatic cell behaves like, or now the somatic cell is like embryonic cell. Means somatic cell is like pluripotent cell. Okay. Therefore, we call it as induced pluripotent precursor stem cells. Okay. Induced. Induced means stimulated. Induced means stimulated. So, induced pluripotent precursor stem cells are nothing but the somatic cells converted to pluripotent cells. Is it clear? It means they are not, iPSCs are not original stem cells, but they are converted stem cells. Is it clear? Because I don't have original stem cells, I take a somatic cell and convert it into a stem cell. Clear till now? Because we do not have, as I told you, I am an adult. So today I don't have my egg cell. Okay, totipotent cell egg cells, it's not zygote. Totipotent cell zygote is not there. The embryo grown into you, you don't have your embryo today. You don't have your zygote today. You don't have your umbilical cord today because it was thrown. You have your bone marrow cells, but they cannot be used to make organs. So if you want to make an organ, for example, if I want a heart, what I can do is I will take my skin cell, convert it into pluripotent, then from that manufacture the heart. I repeat, today if I want a heart, okay, another copy of my heart, then what I can do, take the skin cell, inject embryonic gene transcription factors into it, convert it into a pluripotent cell. From that you manufacture the heart and then surgically arrange it in my body. Will it perfectly match with my body? Yes, because it is made from my somatic cells. It will 100% match with my body. That is the purpose of the iPSCs. Is it clear? Yes, Avinash, because now if the iPSC technology is available, preserving the umbilical cord stem cells is obsolete. It means old style. We need not preserve the umbilical cord stem cells now. But if the iPSC technology is available, but iPSC technology is still not much available to everybody. So still, okay, preserving the umbilical cord stem cells is relevant for a few more years. Is that clear? So, induced pluripotent precursor stem cells are not original pluripotent, they are duplicate pluripotent, they are converted pluripotent. A somatic cell converted into a skin cell converted into a pluripotent cell. Okay? This is the only way by which adults can have stem cells. Okay? Because before birth, if you want, you have zygote. Before birth, if I want to collect something, I have embryo. Soon after the birth, if you want to collect, you have umbilical cord. But if an adult wants an organ whose umbilical cord stem cells are not preserved, my umbilical cord stem cells were not preserved when I was born. So today if I want a heart, then iPSC technology is the only option we have. Because it is having such enormous applications, this particular topic received Nobel Prize for the year 2012. 2012 Nobel Prize, ma'am, it's an old topic. No, Nobel Prize is old, but iPSC technology and stem cell technology are constantly in the news. Is it clear? If you don't want to remember the old topics, just skip this Nobel Prize point. The topic technology, iPSC technology is still important and it is still relevant. Okay. Next topic is 3D organ printing. But before that, let us take a small break. Okay. So, a break for how much time you may need? Break for 20 minutes. -20 minutes break. Okay. So, we will join back by 8:45. Okay, we'll join back by 8:45 p.m. Right? Thank you. Am I audible, visible? Am I audible, visible? Okay. Right. So, welcome back after the break. So, let us now continue with the next topic, that is printable organs, or the topic is 3D organ printing. Okay. Now, the topic is 3D organ printing. Okay. In the last topic we have seen the raw material that are used to make an organ. The raw material that are used to make an organ. Now the topic is using that raw material, how to manufacture an organ. Is that clear? So, for 3D printing, the data about the organ should be obtained by CT scan or MRI. Step 1. First of all, let us suppose I want to print out a kidney. First step I have to do is take the CT scan or MRI of that kidney. Is the step 1 clear? Then step 2. Using that data, make a digital model of that organ. Make a digital model of, it means make a virtual kidney. A virtual 3-dimensional kidney using the softwares like AutoCAD software. CAD means computer aided designing software. Using CAD software, digitize that 3-dimensional image of your kidney. Step 3, okay, printer cartridge. Means what? Just like we have a refill in a pen, every printer will have a printer cartridge. In a normal, in your office, the printer you have, in the cartridge you have black color ink. In the colored cartridge you will have different colors of the ink. But this is a 3D printer. In a 3D printer, the cartridge can be filled. The cartridge can be filled with, okay, either you can fill it with a cake batter, so that we can print out nice designer cakes, or we can print out a cookie batter, so that we can print out some designer cookies, bakery items. Or you can print out some plastic, you can fill some plastic in that cartridge. What am I filling the cartridge with? In the cartridge I am filling the plastic so that I can print out a chair. Or I can fill the cartridge with cement. Fill that cartridge with cement. You can print out a building. Recently a post office was built using the 3D printing. And also in the regular house construction also they are using the 3D printing instead of using concrete manually. So they just print out the complete home. Or you can fill some different material to even you can print out. You can print out an entire rocket engine. For space technology, you can print out a rocket engine for space technology. Same way you can fill the cartridge with cells to print out the organ. Is that clear? In a normal printer we put ink, but in the 3D printer we can keep a cake batter, plastic, cement, or even the cells can be kept to print out an organ. This organ will be surgically transplanted. This organ will be surgically transplanted into the patient. Then it is called 3D organ printing. Okay. Now we keep the cells okay, but for the cells you also need a medium. You also need some medium. That medium is nothing but a hydrogel. Hydrogel. Hydrogel means what? A water-based gel. Okay. Made up of alginate and fibrin. You must be thinking what alginate and fibrin will do in our body. What is the relation between alginate and fibrin in manufacturing the kidney? Nothing. This alginate and fibrin, what they do is, they help in the cell division. Is it clear? They help in the cell division. Cell differentiation. I mean cells becoming into different types of the cells. And also cellular adhesion. Because can I say that a loose group of some kidney cells, can I call it as a kidney? Or these kidney cells should tightly attach with each other to form the organ? These cells should adhere to each other. They should join, they should combine with each other to form the kidney. So it means we need three things. Okay, 1, cell division. 2, cellular differentiation. Cellular differentiation. 3, cellular adhesion. And this alginate and fibrin. Alginate and fibrin helps in all these three things. Okay, means this alginate and fibrin, this is like the medium. To this medium we add the cells, okay, cells of interest. Cells of interest. What do you mean by cells of interest? For example, I want to manufacture kidney. Then the cells of interest are kidney cells. If I want to manufacture heart, then the cells of interest are heart cells. Is it clear? It means cells of interest means cells of whatever organ you want to manufacture. You want to manufacture kidney, then kidney cells. Okay, those are called cells of interest. Now let us suppose a person's kidney is completely failed. You don't have the cells of interest. I mean, you don't have at least a few healthy kidney cells from which you can manufacture another kidney. So if the cells of interest are not available, you can also use stem cells. Okay, which stem cells? That umbilical cord stem cells. Is it clear? Umbilical cord stem cells. Okay, so to manufacture a new kidney, in what situations I can use kidney cells? If that person have at least a few healthy kidney cells, then take those cells of interest. I mean, take the kidney cells. Okay, this person's kidney completely failed. You don't even have a few healthy kidney cells. Then what you can do? Umbilical cord stem cells. But if this person's umbilical cord stem cells were not preserved, then what you can do? iPSCs. So these are the three alternatives you have. You mix these cells into that medium. Together it is called as bio-ink. Bio-ink. It is not actually an ink. Okay, but in the normal cartridge we fill ink. In this we are keeping it here. So symbolically we are calling it as the bio-ink. Okay, what is the bio-ink composition? All of you? The water-based gel, hydrogel, the water-based gel of alginate and or fibrin, water-based gel of alginate or fibrin as the medium. To that you add either the cells of interest, I mean whatever organ you want to manufacture, those cells, or umbilical cord stem cells if they are available. If they are also not available, iPSC cells. So these cells plus that medium together is called bio-ink. This bio-ink is filled in the cartridge. The bio-ink is filled in the cartridge. Then before that, if you remember, we already made the digital model of the kidney is given as the image. Then what happens? The cartridge will print out the real kidney. It's not kidney image, a real kidney will be printed out, and this kidney can be surgically transplanted. The kidney can be surgically transplanted into the patient. This is called 3D organ printing. Is that clear? Then after 3D organ printing, we have this previous year question. 3D printing has applications in which of the following? Okay, actually all the above. We can make cookies and cakes by filling the cake batter into the printer. And we can manufacture bionic ears. Okay, real ears can be manufactured. In the automobile industry, for example, if you design a new car, that car's model can be printed out using this. Reconstructive surgeries. For example, sometimes you get any fracture, femur bone fracture. You have to keep a rod there. Such rods can be printed out using the 3D printing. Data processing technologies like pen drives or processors can be printed out using 3D printing technology. Plus we can also, they have given 5, plus we can also use them in the space technology. Okay, we can print out the rocket. Okay, we can print out a complete rocket. And we can also use it in the 3D organ printing. Biological organs can be printed out. 3D organ printing. So these are the various applications of 3D printing. Is it clear? Yes. Chip manufacturing can be done. Now, Organovo company produced the human liver. As of now, what all organs are manufactured using the 3D printing? Liver, heart and ears. But only ears are successfully transplanted into the patients. Liver and heart were just printed out but not transplanted into the patients. Okay, because those are not up to the mark. I mean, we need more improvisation. Okay, liver and heart were manufactured, but they are not to the level of being transplanted. Okay, but ears are already transplanted using 3D printing. We have printed out the ears and then surgically transplanted them to the patients. Clear till now? Then, now, so this is one way of manufacturing the organs. It means on the whole we have seen the stem cell technology. What is the stem cell technology? These are the raw material. Then after discussing the stem cell technology, we have the next topic, that is how to make the organs. Okay, one way is we can manufacture organs in vitro, means in the laboratory. How? That is nothing but 3D printing. 3D printing of the organs. The other way is in-vivo manufacture. We can also manufacture the organs in-vivo. How? Okay, in the animal embryos. In the animal embryos. Is that clear? Okay. Now, Japan, now this is already completed. Stem cells we have completed. Manufacturing the organs in vitro, that is 3D printing, completed. Now let us see in vivo organ production, that is manufacturing the organs in the animal embryos. Okay. Japan recently approved first human animal embryo experiments. So here what they have done is, look at this diagram. Okay, what are these white color cells here? What are they? They are nothing but the animal embryo, goat embryo or pig embryo. Is the step 1 clear? We have taken the pig embryo here or goat embryo. Done. Step 2. Into that, okay, into that we are adding the human iPSCs. Human iPSC means induced pluripotent precursor stem cells. Got it? And these iPSCs are already treated with the liver specific hormones. This will give you an answer to the question that stem cells can form various organs, but how do the stem cell know which organ they should form now? For example, I am a heart patient, I want a new heart, okay, and I am trying to manufacture a heart. Finally, if it turns out to be a kidney, is that okay? So did you get this doubt? How do the stem cells know into which organ they should get converted into? Is the question clear? Yes, for that what we have to do is, we should take the stem cell and add some specific hormones to it. So if you take stem cells and add liver specific hormones, then the cells know that they should form the liver, they will not form the other organs. So these are human iPSCs treated with liver specific hormones. It means now they can get converted into the liver, they can grow into the liver or they can form the liver. Is that clear? Then implant this embryo into the animal womb, pig womb or goat womb. Okay. Finally, that animal young one will be born with the human liver. The pig baby, the piglet or the lamb will be born with the human liver. Why human liver? Because we get human iPSCs into the goat embryo which are treated with the liver specific hormones. These human iPSCs formed the human liver. Okay, goat baby born with the human liver. Is the procedure clear? Right? Okay. So take the animal embryo, into that add iPSCs, human iPSCs which are already treated with the liver specific hormones. It means now the iPSCs know that they should form a liver. Then inject this embryo into the animal womb. Animal young one will be born with the human liver. So will it lead to the death of that animal? Should we kill that animal to take your liver? Yes, it will lead to the death of the animal. Then is it a violation to the animal rights? Yes, it is a violation to the animal rights. That is the reason why till the year 2019 it was not permitted. Only in the year 2019 in Japan it is permitted. Even today it is not permitted in several countries. Is that clear? Then will the human immune system think that it is goat liver and produce antibodies for it? No, because they are made from, the liver is made from human iPSCs. Okay, will the goat immune system think that it is human liver and destroy that? No, because these cells are there since the early stages of the development. The immune system during the development of the immune system is constantly exposed to these cells. So the goat immune system will think that these are goat cells only because they are present since the very young age. These are all the questions possible. Is it clear? Yes? Clear till now? Then I request you all to completely focus on the class. It's a nice opportunity for you to learn all the subjects in a few classes, like 2-3 classes. Okay, and if needed you can even like completely focus on the class, right? So I hope it is clear. Okay, first question, will it match with my body? Will it match with my body? Yes, because it is made from my iPSCs. Will my immune system reject it? No, because it is made from my iPSCs. Will the goat immune system reject it? No, because it is there since the early stages of development. Should you have to kill that animal to get this liver? Yes, this leads to the death of the animal. Is it against your animal rights? Of course it is against animal rights. Is it clear? Then next one, will it be the size of the human liver? No, but you can grow it in the laboratory to the human size. That's not a problem. Okay, I hope this procedure and all the possible questions are cleared. Okay, now a man gets a pig's heart. Is it the same as what we have discussed previously? Growing the human heart in the animal embryo, is it same as taking the animal's heart and transplanting it into your body? No, they both are different. In this experiment what we tried is, we tried to grow the human liver in the pig womb. But here we are not making the human liver in the pig womb. We are transplanting the pig liver into the humans directly. So this is a different technique. Is it clear? Clear till now? Complete focus please. So, okay, so here what they have done is, they have taken the pig's heart. But if I directly transplant pig's heart into the human heart, if we directly transplant pig's heart into the human heart, okay, will the human immune system reject it? If I directly transplant animal organ, pig's heart into the human heart, will the human immune system reject it? Yes, everybody, fast. It will reject. So what they have done is, in a hospital in Maryland, USA, a doctor transplanted the heart from the pig that had undergone some modifications. They made 10 modifications. Simple. They have taken the pig heart. First step. Then what they have done is, they have inactivated the pig genes. They have inactivated the pig genes. Third step, they inserted the human genes. They inserted the human genes. Now the pig heart resemble human heart. Now the pig heart resemble the human heart. This pig heart is transplanted into the humans. Is that clear? So very simple. Okay, very simple. Take the pig heart, silence the pig genes, add the human genes. Now the heart looks like human heart. So there is less chance that the human immune system will reject it. But still, it's not exactly like human heart, and it rejected and the person died unfortunately. Unfortunately, the man who took the pig heart transplantation passed away. He could not survive with that transplantation. Is that clear? Then, now the next topic is called organoids. What do you mean by? Use the word organoids once. What do you mean by organoids? Organoids means, okay, they are like miniature organs. Organoids means miniature organs. Means this much brain, this much kidney, a small kidney, a small brain, those are called organoids. Then you might wonder, why do we make organoids? Can I transplant this much brain into a person's head? No, useless. Can we transplant this much kidney into a human body? Useless. What you will do with them? These organoids are not for the organ transplantation, rather they are to study the anatomy of the organs. If a doctor, if a medical graduate want to study how the organs are formed, how do they develop, how do they work, they can use these miniature organs. Okay, so they will give a detailed view of the how organ forms and grow. They will give a detailed view about how an organ forms and grows. Next, they provide insights into the diseases. For example, during COVID-19, they understood how the coronavirus will destroy the lungs using a miniature lung. During COVID-19, they understood how the coronavirus destroys the lung cells using the miniature lung organoid. Is it clear? Okay. Study how the drugs interact with organs. For example, I want to study how a particular medicine will influence the liver. Then I can put that medicine on the liver organoid so that I will understand how this drug will influence the liver. Then scientists gained insights into the COVID-19 through miniature lungs, okay, to understand why some people are suffering from long COVID while other people had very minute symptoms for during the COVID-19. Is it clear? Okay, it means why COVID-19 became very severe in few people, not at all severe in few people. What is the difference? How it is affecting the lungs? They understood all these things using the lung organoids or the miniature lungs. So these miniature organs or organoids are not for the transplantation, rather they are for research and understanding the organs mechanism, anatomy, how they form, how they develop and all those things. Is it clear? Okay. Then now moving on to the next topic, that is Human Genome Project. So first, what is a genome? Okay, genome means Saurabh, it is produced using stem cells. Yes, organoids are also produced using the stem cells and 3D printing technology. Raw material is stem cells. The technology used is 3D printing. Okay, but we print out miniature organs just for the research purpose, not for the treatment purpose. Okay, good question, Saurabh. Thank you. So, Human Genome Project. To understand the Human Genome Project, we should first know what is a genome. Genome is the haploid genetic content of a cell. Haploid genetic content of a cell means what? One copy. Haploid means half the number of ploids. We have two diploids. Sorry, we have two ploids. Means half of them is one. So, one copy of the total genetic content of a cell. So, if this definition is clear, can you all tell me one genome is equal to how many chromosomes? One genome is equal to how many chromosomes? First? Yes, exactly. Very good. One genome is equal to 23 chromosomes. In that case, how many genomes you have per one cell? How many genomes you have per one cell? Very good. 2 genomes. Then next question. Is the term genome almost like the term ploid? Genome is also 23 chromosomes. Ploid is also 23 chromosomes. Yes, so that is the meaning of the word genome. Fine. What is Human Genome Project? Project means what? A project work means what? A task. A task to be completed. What is the target here? Task is to know the nucleotide sequence of entire genome. It means in all our 23 chromosomes, what is the ATGC sequence? ATGCs are arranged in which order? Okay, that is the task of the Human Genome Project. Is the task clear? Sequencing the complete genome. How is it sequenced? What is the equipment used? The equipment is a DNA sequencer. What is it? It is like a scanner. Now look at here, like this. What it will do? It will scan. For example, imagine this pen is a scanner. It will scan the first letter and identify that it is adenine. Then scan the second letter. Okay, then scan like that. It will scan each letter and identify what it is. It will scan each letter and identify what it is. Like that, we will come to know the nucleotide sequence present in all our 23 chromosomes. Like that, we will understand the entire genome's sequence. Okay, is the procedure clear? Yes. Then, what are our observations? Observations means what? After knowing the complete nucleotide sequence of our genome, what did we understand? First thing we understood is more than 95% of the genome contains non-coding DNA or junk DNA or selfish DNA. It is called junk DNA because it is not producing the proteins. For the same reason, it is also called non-coding DNA because it is not coding for the proteins. It is also called selfish DNA because it is there in my body but not helping me by producing the proteins. Means making a selfish existence in my body. So we call it as selfish DNA. But interestingly, all these are misnomers. Is it really junk DNA? Is it really junk DNA? No. It has several functions. Is it really selfish DNA? No, it is helping us in several ways. But is it non-coding? Correct. Non-coding is correct because it will not produce proteins. It will not produce proteins. But it will help us in the other ways. So that is the reason why we have 95%. In fact, not 95%. Close to 99% of our genome is junk DNA. Okay. I said that it will not produce the proteins, but it will help us. Now let us see how it will help us let us see. I will ask a few questions, continue to answer please everybody in the class. Yeah, that is how it helps is our discussion. Let us assume, okay, if junk DNA is absent. If junk DNA is absent. Okay, like it's not absent, it is present. Please notice if at all it is not there, then what is the situation? Okay, then let us suppose a mutation occurred. Okay, what is the chance that the chance of the mutation occurring in the functional DNA? What is the chance of that mutation occurring in functional DNA? Is, yes everybody, 100%. Because total DNA you have as total DNA is only functional DNA. Why total DNA is functional DNA? Because junk DNA is absent. Okay, but if 99% is junk, only 1% is functional DNA. Only 1% is functional DNA. Then what is the chance of the mutation? mutation chance in the functional DNA. Yes, all of you? Mutation chance in the functional DNA is only 1%. Okay, fine. Tell me which mutations are more harmful. Mutations in the junk DNA are more harmful, or mutations in the functional DNA are more harmful? Mutations in the junk DNA more harmful, or mutations in the functional DNA more harmful? Okay, mutations in the functional DNA are harmful. Why? Because they cause diseases. They cause diseases. So now if you have junk DNA, only 1% of the mutations in these situations, only 1% of the mutations cause diseases. Remaining all mutations do not cause diseases. Is it clear? So that is the reason why. Now tell me, is junk DNA really junk, or is it very much needed? What the junk DNA is doing? The junk DNA reduce the probability of the genes getting mutated. Junk DNA reduces the probability of the genes getting mutated. Not only that, okay, look at here, there is another very important function. Okay, junk DNA gets mutated. See, if a gene gets mutated, can it become useless? Quickly. If a gene gets mutated, can it become the junk? Gene can become junk by mutation. Same way, can junk DNA form the gene due to the mutations? If the junk DNA, I mean, can a useless person slightly change to become useful? Can the junk DNA change to become a gene? Yes. So junk DNA gets mutated, forms new genes. With new genes, you will get origin of new species. Is it clear? Okay. Not only evolution understanding, evolution occurring also. Okay. So like that we can say mutations are the raw material. Mutations are the raw material for evolution. Okay, listen this carefully. Let us suppose there is an ape, like a monkey. In that monkey there is junk DNA. That monkey's junk DNA got mutated and formed new genes. With the new genes, now the monkey became human being. This is how the evolution occurs. Is it clear? So, on the whole, junk DNA is not really junk. Junk DNA is not at all selfish. It is very useful. Just it will not produce the protein. So, the correct word for it is non-coding DNA. Junk and selfish are misnomers. Agreed? And next observation. We have approximately 30,000 genes in the complete genome. Okay, 30,000 genes in the complete genome. Okay, means what? Tell me. Okay, I need a few calculations. 30,000 genes in one genome. So, how many genes will be there on one chromosome? 30,000 genes in one genome. Then how many genes will be there on one chromosome? Simple. Second class mathematics. Genome is equal to 23 chromosomes. So it means you have to do 30,000 divided by 23, that is around 1300 genes on one chromosome. Is that clear? 1300 genes on one chromosome. Okay. Himanshi, genome is equal to 23 chromosomes beta. Okay. So on 23 chromosomes together, if you have 30,000 genes, then one chromosome will have 30,000 by 23, around 1300 genes. Is that clear? Then, now, so with this we understood what is the Human Genome Project is clear. Now let us see what are the human genome projects that are in the news. What are the human genome projects that are in the news? First one, oldest one. The first one is the Human Genome Project. It was done from the year 1990 to 2003. Means 13 years it took to know the nucleotide sequence of our genome. Is it clear? It took 13 years. Done. Okay, then it was done by International Human Genome Sequencing Consortium and Celera Genomics. That is a fact, nothing to understand. Who did this? Which organization did this project work? International Human Genome Sequencing Consortium and Celera Genomics. Okay. Okay, fine. Now I have one question. Do we all have the same genome sequence or we all have a different genome sequence? Do we all have the same genome sequence or a different genome sequence? Yes, everybody? We all have a different human genome sequence. Okay? So, here, whose genome did they sequence? In the Human Genome Project, did they sequence your genome or my genome or some hero's genome? Whose genome did they sequence? In this, they sequenced the genomes of lakhs of people, not one person, lakhs of people from all over the world. It's a worldwide project. Is that clear? Yes, clear for all of you? Then, the second project is 100K Genome Asia. 100K. 100K means what? 1 lakh. 1 lakh genomes of only the Asians sequenced. In that 50,000 genomes are that of the Indians. Clear till now? Then, next project is Indigen project. It is by the Institute of Genomics and Integrative Biology, IGBI Delhi, and CCMB Hyderabad did this project. Here, they only sequenced the genomes of only the Indians. Is it clear? They sequenced the genomes of only Indians. Genome India project. In that also we sequenced the genomes of only Indians. This project was launched in 2020. The target is sequencing 10,000 genomes of the Indians. Okay. Next one. Telomere to telomere consortium. What is a telomere? Look at the chromosome structure. In a chromosome structure, if I draw an imaginary line here, this left side one and this right side one, what is that called as? That is called as a chromatid, and this is called as centromere. Centromere. This is where the two chromatids join together. Is that clear? And these are the tips of the chromosomes, they are called as telomeres. Telo means tail, last part. Telo means end part. Is it clear? Now telomere to telomere consortium means what? Like for example, I say that you have to read this textbook from cover to cover. I mean first to the last. Okay, top to toe, you have to sequence the nucleotides of an entire chromosome. Like that, okay, it is gapless assemblies for all 22 chromosomes and X chromosome. Okay, but as of now, we have completed this project only for the 13th chromosome. With this point, you may get a doubt. Ma'am, in that case, then the Human Genome Project, Genome Asia 100K, Indigen Project, Genome India Project, in that are we not sequencing the chromosomes from the top to toe? No. Okay. We left some junk DNA. We focused more on the functional DNA. So next level what we have to do, because we know that the junk DNA is also functional. So without gaps you want to sequence the entire chromosomes. Is that clear? Okay. In the Human Genome Project, Genome Asia 100K, Indigen, Genome India, we sequenced the genomes but not completely. We have done it with a focus on only the genes. But telomere to telomere means not only genes, junk DNA, every sequence of the chromosome must be sequenced. Okay. We have done like that only for 13th chromosome, but the target is to do like that for all the chromosomes. Now, till now what all sequences? Okay, fine. Answer a few questions. What is the world's first Human Genome Project? Human Genome Project. Okay. What is the name of the genome project in which only Asian genomes sequenced? Genome Asia 100K. What are the two projects in which only Indian genomes sequenced? Genome India, Indigen Project. Name the project in which a chromosome will be sequenced from one end to the other end, telomere to telomere. All these come under the category called as HGP read. HGP read means, okay, this is nothing but knowing or determining the nucleotide sequence of an existing genome. Is it clear? Genome is already there. I am just reading it. I am just trying to know the nucleotide sequence of that. Those are called as, okay, those are called as the HGP read type of the projects. Is it clear? Clear till now? Okay. Then what is HGP write? What is HGP write? This is not knowing the nucleotide sequence of an existing genome. This is constructing or manufacturing. Manufacturing a genome in the laboratory. Okay. HGP read means what? You are not creating a genome. Genome is there. You are just trying to know the nucleotide sequence. In HGP write, genome is not there. You are creating it. Okay. In the laboratory, you are synthesizing a genome. Then you ask me, can we do that? Yes, we can do that. We can synthesize the complete human genome. That is called as Human Genome Project. Another similar project is called SynHG. From the name itself, it is evident that SynHG means Synthetic Human Genome Project. a major UK-led initiative to create the first truly synthetic human genome from the scratch. From the scratch means from the beginning, using the raw chemicals. What are the raw chemicals? ATGCs. Use ATGCs to synthesize the complete genome from the scratch. That is called as SynHG project. Clear till now? Finally, human genome project completed, human genome studies completed. What are the applications of the genome studies? First application is pharmacogenomics. Did you notice sometimes what happens is same medicine, if you give to some 4-5 people, on one person it will act very well, on the other person it will not act, no impact. On the other person it will show negative impact. Do you agree? Medicine is the same. You are giving to different people. The reaction is not the same. What do you think is the reason why people respond differently to the same medicine? Because our genomes are different. Yes, clear? Our genomes are different. In that case, is it a good way to sequence? For example, I am a doctor, you are like some patient came. What I will do is, okay, I will read his genome sequence entirely. I will read his genome sequence and give a medicine which is suitable to this particular sequence. Means personal medicines, designer medicines. That branch of study is called pharmacogenomics. Is that clear? It's not like one size fit for all. I'm not trying to give the same medicine to everybody. I will read the genome sequence and give a suitable medicine so that he will not have any negative reactions, so that the medicine will be more effective on this person. Okay. To prescribe a medicine according to your genome, do I need the genome sequence knowledge? Without having knowledge about the human genome sequence, can I prescribe a medicine which is suitable to your genome? Is that clear? If I don't have a knowledge about the genome sequence, can we design a medicine for the a particular patient? Not possible. So therefore, human genome sequencing, okay, application is pharmacogenomics. It means if I know the complete human genome sequence, what is the use? I can give designer personalized medicines. If I have knowledge about human genome sequencing, what we can do? We can give personalized medicines, pharmacogenomics. Same way, we can also do gene cloning, gene therapy, genetic engineering, DNA testing, designer baby. All these things are also applications of the human genome project. It means without having knowledge on the human genome sequence, we cannot do all these techniques. But you may ask me, ma'am, what are these techniques? That is our next topic. It's like a checklist. Now factually remember that these are the applications of the human genome project. Each word we are going to discuss now. Done? Clear till now? Complete focus on the class, please. Now, but before we go into those topics, we have other genome studies also. Till now we completed the genome studies of which organism? Human Genome Project, HGP, right? SynHG, telomere to telomere, Genome Asia 100K, Indigen, Genome India. These are all the studies of whose genome? Everybody fast? Only the human. But look at this. This is Earth Biogenome Project. Here the target is to sequence the genomes of all the eukaryotic organisms living on this earth. Humans, all animals, all plants, all worms, insects, cockroaches, ants, mosquitoes. Okay? Sequencing the genomes of all the living organisms on the earth is called as Earth Biogenome Project. It will be useful for maintaining the ecological balance and what is the relation between the organisms. Okay, what are the uses of a particular species to us? This kind of studies will be, and we can also understand about the evolution. Who is our close ancestor? Who is our distant ancestor? Which organism evolved into which organism? These connections between different organisms can be understood. That is the application of the Earth Biogenome Project. Clear till now? Right? Keep responding, please. Now, so till now we have studied the sequencing of the human genome, then sequencing of the all living organisms on the earth. Now let us see genome sequencing of, okay, sequencing the, and one request all of you, as I already said, you don't try to make a running notes of this. You have the notes, you can download this PDF. Second thing, you keep responding, I mean keep saying yes, no, whether you are understanding or not. With that what happens, your focus will be retained. Okay, and because I will come to know that you have understood, I can quickly proceed to the next topic. Otherwise I can repeat something. So keep responding. Done. So now the next topic is sequencing the genomes of pathogens. Sequencing the genomes of pathogens. Okay. Pathogens means what? Pathogens means what? Disease causing substances. Is it clear? Pathogens means disease causing agents. Can be viruses, like viruses, bacteria, etc. Fine. Now tell me, what is the advantage of sequencing the genomes of the pathogens? First thing, diagnosis. Diagnosis. For example, let us suppose you have taken my throat swab. You have taken some sample from my throat. Okay. And in that sample, in my sample, you have coronavirus genetic material sequence. You found the genome sequence of coronavirus in my sample. What does it mean? It means that I got infected by the coronavirus. Otherwise, why do I have the coronavirus genome sequence in my sample? Same way, if a person's blood sample is showing tuberculosis bacterium DNA sequence, I found the TB bacterium sequence in this sample. Means this person is infected by TB. Is it clear? So we can do sequencing of the pathogens, genome sequencing of the pathogens. One is for the diagnostic purpose. Two is to make vaccines, to make RNA vaccines, DNA vaccines, etc. To make RNA vaccines and DNA vaccines, okay, we can use the genome sequencing. Okay. Now, first what we have to do in the diagnosis, what are the different steps? First, grow the culture, grow the bacteria or grow the viruses. Then extract their DNA. Then you perform PCR. Fine. What is PCR? PCR means polymerase chain reaction. Polymerase chain reaction. It is like in vitro DNA replication. Is it clear? In vitro DNA replication. In vitro means what? In the laboratory, replicate its DNA. Okay? And then sequence the DNA or RNA, whatever that organism is having, sequence its DNA or RNA to identify which organism it is. This is how we can use it for the diagnosis. Now same way we have an organization or a group of laboratories called as Global Initiative on Sharing the All Influenza Data. Global Initiative on Sharing All Influenza Data. In influenza virus, this virus causes flu. Influenza virus causes flu. In the influenza virus, we have different strains. We have bird flu, swine flu, okay, Spanish flu. So we have different variants of the influenza virus. What will be the difference between different variants? Genome sequence is the difference. Is it clear? Different variants have different genome sequence. So we can use genome sequence to keep a track of the variants, to know if any new variant emerged. To know if any new variant emerged, we can use the genome sequencing. Clear for all of you? Right? Then, same way, for SARS coronavirus also we have INSACOG. This INSACOG is a group of laboratories. What they will do? They will keep sequencing the coronaviral genomes and will keep searching if any new variants emerged. Okay, if a new genome sequence is found, it means a new variant of the coronavirus emerged. That is the meaning of it. The next term is biological dark matter or dark genome. This is that part of the genome whose function is not clear. Is biological dark matter same as the junk DNA? No. Junk DNA means the DNA that do not code for the proteins. Biological dark matter means that DNA about which we don't know. It means in the biological dark matter you can have functional DNA. You can also have non-functional DNA. You can have both functional DNA and also non-functional DNA. Both can be there. So biological dark matter is just that DNA about which we do not know. Okay. Now moving on to the next topic, that is genetic engineering. Genetic engineering is a broad topic. Genetic engineering is a very broad topic. Anything you do with the genes comes under this topic. In the genetic engineering, okay, one subtopic is like recombinant DNA technology. Recombinant DNA technology. In this technology, what we do is we mix the DNA of two different organisms. We mix the DNA of two different organisms. Therefore, it is called recombinant DNA. Recombinant DNA means what? A new combination of the DNA. Recombinant DNA means a new combination of the DNA. So in this technique we make a new combination of the DNA. Therefore it is called recombinant DNA. So in the recombinant DNA technology we have topics like, okay, we have topics like gene cloning. Gene cloning is a recombinant DNA technology. Okay. We also have GM crops. Genetically modified crops are also recombinant DNA technology because we mix the DNA of different organisms. Is it clear? Then, okay. In addition to this, here you can also have another topic called gene therapy. Gene therapy comes under genetic engineering but will not come under recombinant DNA technology. Because in the gene therapy, we don't mix the DNA of two organisms. We just do gene editing. We just edit the existing DNA. I take human DNA and will do some editing to that. Okay. Then like this, in this only we can also call this as designer baby. Designer baby concept. Okay. So on the whole, genetic engineering is a very broad concept. Anything you do with the genes comes under that. Recombinant DNA technology is any technology in which we combine the DNA of two different organisms. Gene cloning, GM crops both come under recombinant DNA technology. Gene therapy comes under genetic engineering but not recombinant DNA technology because we don't mix the DNA of two different organisms here. Clear till this point? Then, now as of now I explained the meaning of only these two words, genetic engineering and recombinant DNA technology. Okay, you should still understand the meaning of this GM crops, gene cloning, gene therapy. These three concepts we have to now understand. Okay, first let us try to complete the gene cloning procedure. Can we proceed? The first topic we are now discussing is gene cloning procedure. In the gene cloning procedure also, okay, I will explain it in the step-by-step manner. Okay, make sure that each step is clear. Last step is completed, you will understand what is gene cloning. Okay, right. Look at this diagram, but don't look at this diagram like this. Okay, focus on this diagram at the place where I am highlighting. Okay, tell me what is this? It's a human cell. Inside that I have drawn a thread-like structure. What is that? Human DNA. Human DNA. On that there is one gene called as insulin gene. Okay, do we have only insulin gene on the human DNA? No. We have 30,000 genes on the human DNA. But now I cannot show all the 30,000 genes here. So I am showing only that gene about which we are talking. Is it clear? Right now our topic is about insulin gene. I hope this is clear for all of you. The circled part is clear. Now leave that and focus on this part. Only focus on the circled part. What cell is it? It's a bacterial cell. What is this thread-like structure? It is the bacterial chromosome. In addition to bacterial chromosome, what you have here? Plasmid. What is a plasmid? Plasmid You're able to hear me now? Am I audible? Okay, thank you. So now focus on this. It's a combination of the bacterial plasmid. It's a combination of the bacterial plasmid and the human insulin gene, right? Bacterial plasmid and the human insulin gene. Can I call this as a recombinant DNA now? Can I call it as a recombinant DNA? This is called recombinant DNA. Why that is called recombinant DNA? Why that is called as recombinant DNA? Because it's a combination of two different DNAs. It is a combination of two different DNAs. Is it clear? Okay, right. Then now what we do is, so here what we are doing is in the step 1, cut the insulin gene, right? Step 2, imagine this is the plasmid. Okay, it's a plasmid, imagine. Okay, just wait 1 second, I'll take a tool to explain this to you. Okay. So let us suppose that this is your insulin gene. Is it clear? It's your insulin gene. And imagine this circle is a bacterial plasmid. Clear? Keep responding, please. The circle is a bacterial plasmid. Okay? So first what you have to do? Cut the insulin gene. Cut the insulin gene. Then cut the plasmid. Then attach the insulin gene to the plasmid. Then you will get, what is this circle called as? Recombinant DNA. This circle is now called as recombinant DNA. Then place that recombinant DNA into the bacterial cell. Look at here. Place that recombinant DNA into the bacterial cell. Focus here. What is this? This is nothing but that recombinant DNA. Now it is a bacteria having the recombinant DNA. Therefore it is called recombinant bacteria with the human insulin gene. Clear? This is now a recombinant bacteria with the human insulin gene. Clear? Then it means what you have done? Take out this plasmid, add the gene, keep it back. Simple 3 steps. Take out this plasmid, add the gene, keep it back inside the bacteria. Got it? You got a recombinant bacteria right now. Then let this bacteria perform the cell division. The bacteria will undergo cell division. Means one cell will become 2, 4, 16, 32, 64, 128, 256, 1 million, 2 million, 4 million, 8 million. Like that you will get millions of bacteria. Is it clear? You get millions of bacteria. Now tell me all of you, before the cell division, how many copies of the insulin gene we had? Before cell division, how many copies of the insulin gene we had? Only 1 copy. But after the cell division, how many copies of the insulin gene we had? 1, 2, 3, 4, 5, 6, millions of copies. After the cell division, you got millions of copies of the insulin gene. Now you got one question. Why did we transfer human insulin gene into the bacteria? Because we are using the bacteria like a Xerox machine, okay, to copy our insulin gene. Is it clear? Copy our insulin gene. Now, the bacteria will do 2 things. The bacteria will first multiply the gene. 2, it will also produce insulin protein from the insulin gene. So the bacteria will do 2 things for us. First, it will increase the number of copies of the insulin gene. Plus, it will also produce the insulin protein from the insulin gene. Is that clear? Then, next what we have to do? Okay, extract the insulin protein from the bacteria. Extract insulin protein from the bacteria. Isolate and purify and package the insulin, and then it can be used as a drug to treat diabetes. I hope you all now understand why did we keep the human insulin gene in the bacteria. The bacteria will multiply the human insulin gene. Plus it will also produce the insulin protein. This insulin protein can be extracted from the bacteria, isolate it, purify that, package it in the vials, send it to the pharmacies. Patients can purchase this and use it as an injection for diabetes. That is the purpose of the gene cloning. Done. Then you may get one doubt. Is the gene cloning only for diabetes? No. Look at here. For example, in the place of this growth hormone gene, okay, then you will get a growth hormone gene. Got it? Growth hormone. Then what happens? You got recombinant bacteria with human growth hormone gene. Then the bacteria will multiply the gene and will also produce the growth hormone. Then extract the growth hormone protein. Extract the growth hormone protein. Isolate it, purify and use it to treat dwarfism. Is the point clear? So it is not insulin gene. Better word is gene of interest. We better call it as gene of interest. Whatever protein you want to manufacture. Is that clear? Like that you can produce any things like say clotting factor, blood clotting factor. Then it will be used to cure the hemophilia. Hemophilia is a disease where our blood will not clot. Even in the case of a small injury, the person can bleed till the death also. So what you can do is clotting factor number 8 is missing in them. So clotting factor 8 gene, okay, kept it in the bacteria. Clotting factor number 8. Okay, clotting factor number 8. Okay, then the bacteria will copy the clotting factor number 8 gene. Then it will produce the clotting factor number 8. Okay, then if a hemophilia patient is severely bleeding, you can give this clotting factor injection. His bleeding will stop. Okay, like that we can use gene cloning to produce various proteins. Clear till now? Done? Then we can also use gene cloning to produce vaccines also. Can we proceed? Okay. Now the next topic we are discussing is vaccines. First, what do you mean by a vaccine? First, answer this question. Does a vaccine contain, for example, if I take a polio vaccine, does the polio vaccine contain polio virus itself or a medicine for the polio? A vaccine contains the pathogen itself. Then next question. Does a vaccine contain the antigen or the antibody? Vaccine contains the antigen or the antibody? Vaccine contains antigen. Then you may ask me, what is an antigen? Antigen is like any biological material that can trigger any biological material that can trigger the production of antibodies. Okay, please notice I am not saying living material, it can be dead also. Any biological material that can trigger the production of antibodies is called as. It means if it enters into my body, it should be capable of triggering the production of antibodies by my immune system. Then we call it as an antigen. Is the antigen word clear for all of you? Okay. Then what is an antibody? Antibody is a defense molecule produced by the immune system. It's a defense molecule produced by the immune system. Okay, and antigen and antibody are highly specific. It means, can the corona antibody destroy the polio virus? Can the polio antibody destroy the coronavirus? No. Okay, they are like lock and key. Okay, then antibody production is always a reaction. Means we can never produce antibodies before our body is exposed to the antigen. For example, a particular antigen never entered into my body. Can I produce an antibody for that? No. Antibody production is always a reaction. It occurs after our immune system gets exposed to the antigen. Clear till now? Right. Now, so the vaccine contains the pathogen itself. So, you may get a doubt, if the vaccine contains the pathogen itself, is there a possibility that sometimes the vaccine itself can cause the disease? Yes. So, to avoid that, what we do is, first we try to take the killed vaccines. Means take a polio virus, kill it and then inject. Okay. Then what happens? The virus is dead. So it cannot cause polio in my body. But its structure is intact. So it can trigger the production of antibodies. Now my immune system will produce antibodies. Okay. And now if I really get the polio virus, then these antibodies will fight with that. On the whole, what is a vaccine principle? Vaccine principle is like making weapons before the war begins. For example, let us suppose Pakistan started attacking on India, and then can you just order S-400 from Russia and then get it ready? We should be ready with the weapons. So here also, okay, I cannot wait till the real polio virus comes into my body, because if the real polio virus comes into my body, before we produce antibodies, it will destroy my organs. Is it clear? Because antibody production takes some time. It will take some 2 to 3 days for the antibodies to be produced. So meanwhile, the virus can destroy my organs. So what you have to do? Be prepared with the weapons. How? Don't wait for the real polio virus to come. You willingly inject a killed polio virus. Okay. Because it is killed, it cannot cause polio, but it can trigger the antibody production. So, polio antibodies are produced. Now I am ready with the weapons. If the polio virus really comes, these antibodies will immediately destroy that. This is one strategy. Second strategy is we take inactivated. We also call this as live attenuated vaccines. Okay, live attenuated vaccines means what? You are not killing the virus, just inactivate it and inject. Because it is inactive, it will not cause infection. But because its structure is intact, it can trigger the production of antibodies. Antibodies are produced, my weapons are ready. So if the real pathogen comes, I am ready to fight with that. Okay. The third one, now look at this image. In this image, can you tell me the killed vaccines, live attenuated vaccines, both come under, okay, which category? This one, A or B or C? Which image correctly demonstrates the two types of the vaccines that I already explained? A or B or C? Yes? Only A. It is whole virus or whole no, only A. Whole virus, complete virus or complete bacterium, either killed or inactivated. Is that clear? Killed or inactivated, but you are taking the complete whole virus or whole bacterium. Is that clear? Then the third type of vaccines are called as subunit vaccines. Subunit vaccines. In this, only a part of the virus is injected, is given as a vaccine. Is it clear? Only one part of the virus is given as the vaccine. Is that clear? Now, look at here, A, B, C. Which one correctly demonstrates a subunit vaccine? First, A or B or C? Okay, B can be called as subunit vaccine. Subunit vaccine can also be called as recombinant vaccines. Look at here. Subunit vaccine can also be called as recombinant vaccines. Why are they called? No, not C. I will tell you why. Saurabh, your doubt is valid, but I will tell you why. The subunit vaccines are also called recombinant vaccines. Fine. Why are they called recombinant vaccines? Because they are made using the recombinant DNA technology. They are made using the recombinant DNA technology. Means how? Look at here. I will just change everything here. You know, coronavirus will have spike proteins. This is called a spike protein. Spike protein can also be called S protein. And this is called N protein. Is that clear? This one is called N protein. Okay. Inside that you have RNA. It's an RNA virus. It's an RNA virus. On that you have, okay, this is S gene, this is N gene. So don't confuse between gene and protein. Okay, gene and protein are not the same. Remember that gene will produce the protein. So S gene produce S protein. Okay, S is common. Don't focus on S. What is it? It is S gene, it is S protein. Okay, that is the difference. Clear? Now, so what you do is, okay, this S gene produce this S protein, correct? Quickly respond please. This S gene produce this S protein. So what you do is, imagine, okay, now I will change here. Okay, this is not human cell. Now imagine this is coronavirus. In that it is not insulin gene. Imagine it is S gene. Are you all getting this? It is not human insulin gene. It is SARS-CoV-2's. It is SARS-CoV-2's S gene. Is that clear? Then it is recombinant DNA bacteria with SARS-CoV-2's S gene. Okay. So this is S gene. Now what happens? Bacteria will do two things. It will multiply the gene. It will multiply the gene and produce the S protein. It will multiply the S gene and will produce the S protein. Is that clear? And so it will multiply the produce the S protein from the S gene. Is that clear? It will produce the S protein from the S gene. Not visible? Writing is not visible, huh? You're able to see this now? Slide is not changing. Slide is not changing. Is it visible? No. Not changing. Okay. Able to hear me? Am I audible, visible? Okay. So, hope the slide is changing. The slide is not changing. Right. Okay. You are able to see what I am writing? Fine. So, we completed this one. Killed vaccines we have completed. Live attenuated vaccines we completed. Both of them come under the A category. Now, third one is Subunit vaccines, they are called subunit vaccines because we take only one part of the virus, not the complete virus. Subunit vaccines are also called recombinant vaccines because we make them using the recombinant DNA technology. Okay? Clear till now? Right? Why the subunit vaccines are called recombinant vaccines? Because they are made using the recombinant DNA technology. Right? Now, how they are made using the recombinant DNA technology is the point. How they are made using the recombinant DNA technology. Same as the way we manufacture the insulin. How did we manufacture insulin? Now just imagine, this is not human cell. It is a coronavirus. This circle is a coronavirus. Coronaviral genetic material. On that spike protein gene. Okay? Yes? Then, now keep that SARS-CoV-2's spike protein gene in the bacterial plasmid. We got a recombinant DNA. Then place it into the bacteria. You will get a recombinant bacteria having the SARS-CoV-2's spike protein gene. Is it clear? We get a recombinant bacteria with the SARS coronavirus 2's spike protein gene. Now the bacteria will do two things as you already know. It will multiply the spike protein gene. Plus it will produce the spike protein from the spike protein gene. Then you can extract the spike protein. Is that clear? Extract the spike protein. Isolate, purify and package this spike protein. And then use it as a vaccine. This spike protein is used as a vaccine to prevent COVID-19. Is the procedure clear for all of you? It is recombinant DNA. Okay, RNA, right. It is actually RNA. Okay, but roughly to explain the procedure I have given like this. Okay, coronavirus actually contains RNA, but we can convert its RNA into DNA. Okay, note SARS-CoV-2 contains RNA, but it can be converted to DNA. Is that clear? So that gene we insert. Why this is called recombinant DNA technology? Why we call it as recombinant vaccine? Because we made using the recombinant DNA technology. Is the procedure clear? Okay. Then now the next type, fourth type of vaccines are called as RNA and DNA vaccines. RNA and DNA vaccines. Here, okay, not the S protein, rather S gene is given as vaccine. Is that clear? In the subunit vaccine, we give S protein. Now we give S gene. Okay, don't confuse between gene and protein. Gene gives protein. S gene produces protein. S protein is used in subunit vaccine. In the subunit vaccine, we give S protein. Whereas in the RNA or DNA vaccine, we give the S gene. Okay, then who will produce? Then we will produce the S protein and then produce antibodies for S protein. Is the procedure clear? In the subunit vaccine, we inject the gene into the bacteria. The bacteria produced the protein and we inject the S protein. But in the RNA DNA vaccine, we do not inject the S protein. We inject the DNA. It's DNA I will inject it into my body. It's RNA I will inject into my body. Then its spike protein will be produced by my own cells. And then my immune system will produce the antibodies for that spike protein. Then it is called genetic material vaccine. Look at the diagram and tell me, okay, Saurabh, now you tell me, among the three, which diagram best describes the genetic material vaccines? Okay, this is RNA or DNA vaccines. Is that clear? Done? That's all. Now, another diagram for you. Here, this is called viral vector vax. Okay, fine. Let us complete the familiar boxes first. Okay, look at this one. It is weakened or inactivated vaccine. Is this box clear? Weakened or inactivated or attenuated vaccine. Weakened or inactivated or attenuated vaccine. This is like that, this number 1, isn't it? That is like this A, like A. Complete virus, whole virus. Example, Bharat Biotech's Covaxin is a, okay, some of you might have taken Covaxin for coronavirus. Covaxin is a live attenuated vaccine. Is it clear? Covaxin is a live attenuated vaccine. Same way, Sinopharm, Sinovac are also live attenuated vaccines for coronavirus, but we did not use them in India. They are Chinese vaccines. Clear till now? Can I say the first box is completed? Fourth box, sorry. This box is clear? Quick responses, please. This is the subunit vaccine I just told you till now. I mean B. This is like our B. Subunit vaccine are also called as recombinant vaccine. What are we injecting? Spike protein. Who produced that spike protein? Bacteria. What are you injecting into the human body? The spike protein, not the gene. Okay. Example, Corbevax, Covovax, Novavax for coronavirus, Cervarix and Gardasil for human papilloma virus that causes cervical cancer. Okay? Is that clear? Corbevax, what are they? They are just brand names. Corbevax, Covovax, Novavax for coronavirus, Cervarix and Gardasil for human papilloma virus that cause cervical cancer. Is the second box clear? Third box I already told you. Third box is like C. Only the genetic material. If it is an RNA virus, inject its RNA. If it is DNA virus or a bacterium, inject its DNA. And then we produce a spike protein. Who will produce a spike protein? The human cell. Produce the spike protein and then we produce antibodies for it. Example, Pfizer, BioNTech, Moderna are the RNA vaccines. Zygote is a DNA vaccine for coronavirus. Then you may ask me, ma'am, coronavirus is an RNA virus. For it, how can I make DNA vaccine? I told you RNA can be converted into DNA. Is it clear? RNA can be converted into the DNA. Okay. I hope these three boxes are clear. Then the last box, viral vector vaccine. Now let us try to understand what is a viral vector vaccine. Okay. The word vector means vehicle or a carrier, something that will carry. Okay. In making this, we use bacterial plasmid as a vector, isn't it? In the subunit vaccines, we use bacterial plasmid as a vector for S gene. But in the viral vector vaccines, in this box, for this particular box, in viral vector vaccines, we use a viral genome as a vector for, okay, S gene. Is it clear? That viral vector is nothing but ChAd-OX. Okay, you don't have that word. Note down this word, ChAd-OX. You can draw this. This is ChAd-OX1. CH stands for chimpanzee. AD stands for adenovirus. OX stands for some number. Okay, that serial number. Okay, chimpanzee adenovirus DNA used as vector. So what is this? This is chimpanzee's adenovirus DNA used as vector. Vector for what? Vector for, imagine what is this? S gene. Remaining process similar to, okay, this like genetic material vaccine. This combination, so this is also recombinant DNA. This combination is injected into humans as vaccine. Then next, who will produce the protein here? Here also fine. Here also the human cells produce the S protein, and then the immune system produce antibodies for S protein. Is it clear? So what is injected here? I will simply summarize it. Take the chimpanzee adenovirus DNA. To that add the S gene. Then inject this DNA into our body, not protein, DNA into our body. Then our cells will produce the S protein. Then our immune system will produce antibodies for it. We are ready with antibodies. If the coronavirus really comes, we can fight. Is it clear? So now let us summarize. Here what we will inject. Okay, in this case what we will inject? Complete virus. In this case what we will inject? Only the spike protein. In this case what we will inject? The RNA or the DNA alone. In this case what we will inject? It's DNA along with chimpanzee adenovirus DNA. In these two cases who will produce? In these two cases who will produce the proteins? Human cells will produce the proteins. In this case who produced the protein? Bacterial cell produced the protein. Clear? Complete clarity? Only difference is here we are injecting the coronavirus DNA directly. Here we are injecting the coronavirus DNA by keeping it in a vector. Here only, I'll write the difference. Only coronavirus DNA here. Coronavirus S gene plus ChAdOx1 here. Okay, that is the only difference. Done? Clear? Right. So with that we completed the vaccinations story. Solve this question, all of you. In the context of vaccines manufactured to prevent COVID-19 pandemic, consider the following statements. Okay, right. Is it correct? Serum Institute of India produced COVID-19 vaccine named Covishield using mRNA platform. Is Covishield an mRNA vaccine? Is Covishield an RNA vaccine? No. Then what vaccine is it? Covishield is a viral vector vaccine. Okay. Sputnik 5 vaccine is manufactured using vector based Sputnik 5 vector based, correct. Chimpanzee adenovirus vector. Covaxin is inactivated pathogen based vaccine, correct. So 2 and 3B is the right answer. Now, one more thing. In this place of S gene, you can also keep, for example, you can also keep Ebola viral gene or Nipah viral gene. Means vector is the same. You change the gene, you make vaccine for Ebola virus, you can make vaccine for Nipah virus, etc. Is it clear? Same ChAdOx1 can be used as a vector to make different vaccines also. Got it? Then, now the next topic is gene therapy. Gene therapy is of gene cloning. What is the difference? Okay. In gene cloning, fine, answer this question. In gene cloning, we inject the protein. Okay, tell me, in the gene cloning we manufacture the insulin and we inject insulin to cure the diabetes. Is it a one-time solution or should you give the insulin injection regularly? Is it a one-time solution or we should inject the insulin regularly? Yes, everybody? Regularly. Yes, I will give you the break just because now you just asked for the break. We cannot stop in between the topic. Let us complete this topic and then we'll take the break. Okay? Now, complete focus on this. Okay, regularly we have to give. Is it solving the problem at the root cause level? For example, my insulin gene is mutated. I am unable to produce the insulin. Okay, I will take insulin protein as an injection. I will take insulin protein as an injection. Is it a solution at the root cause level or only like a superficial solution? It is only a superficial solution. We are not solving the problem at the root cause level. We are not solving the problem at the root cause level. Is that clear? Then, so what is solution at the root cause level? You should correct the mutation in the gene. You should correct the mutation in the gene, isn't it? Then only it is like solving the problem at the root cause level. Agreed? Clear? Okay. So, means we have to do some gene editing. We have to do some gene editing. Is it clear? Means gene therapy is nothing but remove a mutated gene and then add a correct gene. Remove the mutated gene, add the correct gene. This is called as gene editing or gene therapy. Gene editing or gene therapy. Is it clear? Remove the mutated gene, add the correct gene. That is, by doing that, for example, my insulin gene is mutated. Remove that, add the correct insulin gene. Then what happens? It's a gene therapy. I solved the problem. It is solution at the root cause level. It is a solution at the root cause level. And it is one-time solution. It is one-time solution. Now because you have corrected the gene, I can produce the insulin. You need not give the insulin hormone repeatedly every day. Is it clear? Then, now depending on in which cell you have done this, we have somatic gene therapy. Somatic gene therapy means you corrected the mutation in the somatic cells. Advantage here is there are no legal implications, means it is legally permitted. But drawback is practically very difficult due to large number of cells to be edited. Okay, there are so many cells to be edited. Okay, therefore it is not an easy technique. Okay, for example, you want to do the pancreatic treatment. In the pancreas, you have billions of cells. So can I do this editing billion times? Can we repeat this process billion times? I can remove the wrong gene, keep the right gene, remove the wrong gene, keep the right gene. Can I do that process billions of times? No, that is a problem with the somatic gene therapy. Then, second type of gene therapy is called embryonic gene therapy. Simple. Delete the mutated gene and add the correct gene in the embryonic cell before even the baby is born. So it is not treating the genetic disease, rather it is like preventing the genetic disease before even the baby is born. Is it clear? With this, the limitation is, advantage is it is easier compared to somatic gene therapy because lesser number of cells to be edited. Maybe only 4 times you have to do it, or little later if you do only 8 times you have to do it. Then some of you may ask me, then ma'am, then why don't we do it in the zygote itself, so that we have to do the editing only once. You can do, but zygote is too delicate to handle. Zygote is after all a single cell. Okay. In this procedure, if something wrong happens to it, the zygote may completely die. But embryo, if you take embryo, is a group of cells. Even by mistake, if one cell dies, the other cells are still alive. So it is better we do it in the embryo, not in the zygote, because it is too risky to deal with the zygote. Agreed? Then, drawback is it is not legally permitted. Why it is not legally permitted? It is good only now. We can prevent the genetic disease even before the baby is born. It is an excellent technique, very useful. So why don't we permit it? Because it can lead to designer babies. Designer baby means what? For example, I want my baby to have fair skin, very intelligent, curly hair, blue eyes, okay, very strong, healthy. Like this I can design the baby. If you can delete a mutated gene and keep the correct gene, then same way you can delete dark skin gene to keep the fair skin gene. Same way you can remove the black eye gene and add the blue eye gene. So this kind of fancy ideas the people may get, okay, which is not good, because if the designer baby technique is permitted, then what happens? All the rich kids will be looking beautiful, strong, intelligent, capable. All poor kids will be born less capable, less intelligent, not very good looking, etc. It will become another source of discrimination between the rich and poor. So we should not permit the designer baby. But the gene therapy, embryonic gene therapy can lead to designer baby. So though the embryonic gene therapy is very useful, it is not permitted. Is it clear? Okay. Then now in this CRISPR-Cas9 mechanism, I skipped. I did not explain CRISPR-Cas9 mechanism, but look at here. The CRISPR-Cas9 mechanism and the updates related to the CRISPR-Cas9 mechanism, like BIRSA 101, okay, GLO-Cas9, like that many news articles related to the CRISPR-Cas9 are discussed in our phase 1, and I have provided the link for that particular session here. So click on this link, you will get CRISPR-Cas9 mechanism plus also all the current affairs related to the CRISPR-Cas9 mechanism. Is that clear? Okay. And I have planned like this because instead of repeating the same topic in multiple phases, it is better to cover different topics in different phases so that I can give you more information. What is your, please share your view on this. Same CRISPR-Cas9 I will teach in all the phases will be useless, right? So that's why I just planned it like that. Okay. So in the 5 phases of Conquer Prelims in my subject, I will not repeat the topics. I will plan different topics in different phases of the Conquer Prelims. Okay. Now, what is designer embryo and embryonic gene therapy differences? Simple. We use the word embryonic gene therapy if it use it for medical purpose. We call it as designer baby if you do it for aesthetic purpose, like beautiful baby, like that. In the designer baby, what we do is we first scan the embryonic DNA, means genome studies, completely study the embryonic genome. Then you can identify the mutations and then correct it and then implant that designed embryo into the womb. Baby will be born. I repeat the step. Scan the embryonic DNA. Then you will identify the mutations. Correct them using the gene editing tool like CRISPR-Cas9. Then implant the modified embryo into the womb. Then implant that modified embryo into the womb. A designer baby will be born. Okay. So, again CRISPR-Cas9 won the Nobel Prize for the year 2012. Is it clear? Chemistry Nobel Prize for the year 2012 is for CRISPR-Cas9. Done? So, CRISPR-Cas9 is very much in the news. If you don't want to read old topics, just forget the Nobel Prize point. CRISPR-Cas9 is very much in the news. Then consider the following. Solve this question. Genetic changes can be introduced into the cells, produce egg cells. Don't have to read all these things. Genetic changes can be introduced into any cell. Any cell you can do the editing. Next. A person's genome can be edited before the birth. Embryonic gene therapy. Yes. I will write the answers also here. This is correct. No, sorry. This is correct because any cell can be edited. Next. A person's genome can be edited before the birth. Correct. That is called embryonic gene therapy or designer embryo. Human-induced pluripotent cells, iPSCs, can be injected into the embryo of a pig. Yes, for in vivo organ production. For the in vivo organ production. Is that clear? Next, what is Cas9 protein often used in the news? It is a molecular scissor that can cut the mutated gene, and in the gap we can add the correct gene. Cas9 is a molecular scissor. It can cut the mutated gene. In the gap we can add the correct gene. Is that clear? The next topic will be GM crops. Okay, but meanwhile we can take a small break for 15 minutes break. Let us be back at 11:10. Okay, 11:10 I will be back. Okay. So welcome back after the break. Hope you're able to. Is my audio video clear? Welcome back after the break. So actually I'm planning this class. The class will be till 1 p.m. Then, yeah, the class will be till 1 a.m. Right? And we will be completing only biotechnology in this class. On 13th March, you have another class. In that class, I will cover the nuclear, ICT and nano. Okay. Then after that, towards March ending, you will have one more class. In that, I will cover space technology and defense technology. So it means initially I planned to take it like in 2 sessions, but now I am splitting this into 3 sessions. In today's class, we'll complete biotechnology. 13th March, nuclear, ICT, nano. March ending, one more session will be there. In that class, I will cover space technology and defense technology. So this is our plan. And as I already told you, you can download this PDF in the description below or in our official Telegram channel. In both the places, it will be available. And also, coming to the importance of science and technology. Tell me what is the average number of questions in prelims? Science and technology in prelims, what is the average number of questions? Minimum 11 to maximum 18. So average number of 15 questions are covered from science and technology. Range is from 11 to 18. Average is not 11. Average you will have around 15 questions you can find. So it is comparable to any other subject like some geography or economy or some other subject. Okay. And then in these 6 subjects like biotechnology, nano, nuclear, ICT, space, defense, what is the subject-wise distribution? If I go with around 15 questions, you are getting around 4 from biotechnology, around 4 from ICT, okay, and then or 4 to 5 you are getting from biotechnology, around 4 from ICT, okay. Then remaining you are getting like around one question each like that. Is it clear? So 1 or 2 questions may be from the space technology, 2 questions. So roughly this is the distribution. So from the 6 subjects, the most contributing subject in most of the years is biotechnology. Second rank goes to ICT, computers and information technology. And then remaining subjects are variable. In some years, they are asking more questions from space. In the other years, more from defense. In one more year, more from nano. But it is not standard. But biotechnology and ICT, standardly they are giving some 4-5 questions. So this is the distribution. Is it clear? Now, so after giving this particular information, let us now proceed with the remaining parts in biotechnology. So this class will extend till 1 o'clock. Meanwhile, we will complete biotechnology. So in biotechnology, the next topic is GM crops, genetically modified crops. In the genetically modified crops, in India, the regulation of the GM crops is by GEAC, Genetic Engineering Appraisal Committee. But GEAC will not give permission for the cultivation of the crops. it will only give approval for the cultivation. It means, okay, like it will recommend it to the government. Then if the government gives the approval, then only we can commercially cultivate the GM crops in the country. Is it clear? GEAC cannot approve or cannot permit the commercial cultivation of the GM crops. Okay. So GEAC will give recommendation to the government should approve it. Okay. This GEAC is not a constitutional body, it is an extra constitutional body because it is not incorporated under any article. Rather, it is constituted under the Environment Protection Act 1986. Why this GEAC comes under Environment Protection Act? Because it will also do analysis of whether the GM crops are safe for the environment or not. Not only for the human beings, are they safe for the environment or not is also one aspect. Therefore, it is constituted under the Environment Protection Act. Till now, GEAC recommended only 3 crops in the country: BT cotton, BT brinjal, GM mustard. But out of the 3 recommended, the government approved the commercial cultivation of only the BT cotton. So currently in the country, the only crop which is commercially cultivated, the only GM crop which is commercially cultivated in the country is BT cotton. And the GEAC and these approvals are only related to commercial cultivation. This is not related to import. You can import GM crops into the country, that's a different matter. It will not come under the purview of GEAC. Next, you can cultivate them for research purpose, it will not come under this GEAC. These regulations are only for commercial level cultivation within the country. Is that clear? 3 GM crops are recommended, only 1 GM crop approved, that is nothing but the BT cotton. And how they make this BT cotton, let us try to understand. Okay, look at here. Again focus on this at the place where I am circling. What is it? Bacillus thuringiensis. Bacillus thuringiensis. Bacillus thuringiensis is nothing but a soil bacterium. It's a bacterium that lives in the soil. And then let us assume that this is its DNA. The DNA of the Bacillus thuringiensis. On that there is one gene. This gene is nothing but Cry1AC. Cry1AC. Is that clear? What is Cry1AC? It is a gene that produces BT toxin. Toxin means what? Poison. It's a poison producing gene. It's a poison producing gene. Now this gene is cut from this soil bacterium called Bacillus thuringiensis and then it is transferred into the cotton seed cells. Then you may get a doubt, why seed cell? Why not the plant cell? Same reason like why embryonic gene therapy, why not somatic gene therapy? A plant will have numerous cells, billions of cells will be there. You cannot keep a Cry1AC gene into every cell of a plant. But a seed like an embryo will have limited number of the cells. So we transfer this gene into the cotton seed cells and then a crop is raised. That crop is called BT cotton. Okay. But now comes the question, why we have done this particular procedure? Because we kept a poison producing gene. It means we kept a poison producing gene into cotton plant. Means now the cotton plant is poisonous. So we made a poisonous cotton plant. Why? Because if bollworms eat this cotton, then they will die. If the bollworms eat this cotton, then they will die. Is that clear? It means this particular plant is resistant to the bollworms. Bollworms cannot damage this crop. If they try to eat this plant leaves, then the bollworms will die. So it means the BT cotton is a HYPR variety. HYPR means what? Just keep responding please. HYPR means high yield pest resistant variety. High yield pest resistant variety. Okay. Yield increases because the pest is not damaging the crop. That's all. This is BT cotton. Okay. Then same way we can also make BT brinjal. How do we make BT brinjal? Okay, look at here. Complete focus. How to make BT brinjal. Same thing. Okay. You take Bacillus thuringiensis, which is a soil bacterium. From that Crivinaceae, cut that gene. It's a poison producing gene. Transfer this gene into this time. Okay. Not cotton seed cells, brinjal seed cells. Is that clear? Then you will get BT brinjal. You will get BT brinjal, which is resistant to, this time not bollworms, because bollworms infect the cotton crop. Which worms infect the brinjal crop? Okay, FSB. FSB is nothing but fruit and shoot borer. Fruit and shoot borer. Okay, these are called as Lepidopteran insects. Lepidopteran insects. Lepidopteran insects means what? Insects belonging to the Lepidoptera class. Okay. Lepidoptera is a group of insects belonging to that group. They usually infect the brinjal crop. So we should make a cotton plant resistant to bollworms. We should make a brinjal plant resistant to the fruit and shoot borer and other Lepidopteran insects. Okay. And this Crivinaceae gene produces a toxin which can kill both of them. Not only both of them, it will kill a few insects, not all. It won't kill the butterflies, it won't kill the honeybees, etc. Okay. This toxin selectively kills only a few types of insects or pests like this. So this is the story of BT brinjal and BT cotton. Clear till now? Then advantages. Resistant to the bollworms. It is first advantage. Second one, reduced usage of the pesticides. If the BT cotton, BT brinjal are naturally resistant to the pest, why do we use chemical pesticides on them? It will reduce the usage of the chemical pesticides. Next, results in the improvement of the yield. I told you that they are called HYPR varieties. And then, okay, you may get one doubt. Won't this toxin harm the human beings? Maybe, but we don't eat the cotton. So that problem may not be there. Then you may ask me, okay ma'am, we don't eat, but some animals will graze on the leaves. Then like that it can enter into the food chain. True, but interestingly, this particular toxin will get inactivated in our acidic stomach. Whereas these insects will have basic environment in the insect gut. Is it clear? The insect gut will be basic, pH above 7. So this toxin will get activated only in the basic conditions, will get inactivated in the acidic conditions. So tell me, can this BT toxin harm the human beings and other animals? Quickly. Can the BT toxin harm the humans and other animals? No, because we have acidic gut. Can it kill the insects and worms? Insects, worms and pests? Yes, but not all. Okay, then disadvantages. The BT cotton seeds are more expensive. BT cotton seeds are more expensive. Second one, bollworms are developing resistance to the BT toxin. Bollworms are developing resistance to the BT toxin. And Monsanto company is keeping commercial interest. Okay, what is the commercial interest? Look at here. Imagine this is your Cry1 AC and this is terminator gene. Okay, what the Cry1 AC will do? Cry1 AC will give HYPR, high yield pest resistance. Terminator gene makes the seeds terminator seeds. Terminator seeds. Terminator seeds means what? Single use seeds. For example, if I purchase BT cotton seeds and I will sow them in the field, I will get one crop. Okay. And in that crop I will get the seeds. I cannot use them again to get the second crop. Second time again I have to purchase the seeds. I hope this point is clear for all of you. Normally, a farmer purchases the seeds only for the first time. For the second time onwards, okay, he will get the seeds from the previous crop. But here the seeds we get from the previous crop cannot be used to get the crop once again. So therefore, these are single use genes. Means every time they have to purchase the seeds. This is the commercial interest of Monsanto company. So these are the disadvantages with respect to the BT crops. Okay. BT brinjal also I said. Okay. Resistance against Lepidopteran insects. The same points I said. Lepidopteran, fruit and shoot borer. Okay. Cry1 AC gene into the brinjal variety. Like that. Now the next topic is Dhara mustard hybrid, that is okay, like GM mustard, genetically modified mustard. Okay. Now, so to explain the genetically modified mustard, first of all I have to explain what is pollination. Pollination means what? Pollination means transfer of the pollen grain from the stamen to the pistil. Okay, transfer of the pollen grain from the stamen to the pistil is called as pollination. This is for the sexual reproduction of the organisms, of plants. Is that clear? It is for the sexual reproduction of the plants. Keep responding, please. Right? Here what happens is we have two types of pollination. First one is called as self-pollination. Self-pollination means imagine this is the plant with a flower. Here you have stamen. Here you have pistil. This is the male part. This is the female part. Okay. Then what is transferring from here to here? Something called as a pollen grain is transferring from the stamen to the pistil. Is that clear? Pollen grain transferring from the stamen to the pistil. Clear? This is called self-pollination because this plant is pollinating its own self. Okay? Not getting the pollen grain from the other plant. Is it clear? It is not getting the pollen from other plant. Clear till now? It's not getting the pollen from the other plant. Now the next type is called cross pollination. In cross pollination, what happens is, here you have only the stamen, it is a male plant, you have a female plant separately. Okay, here you have the pistil, this is a female plant. Then here the pollen grain transfers from the male plant to the female plant. This type of pollination is called as cross pollination. Cross pollination, because there is a cross between two different plants. Is that clear for all of you? Then, what do you mean by the word hybridization? Hybridization means mixing two varieties. Mixing two varieties to get a better variety. Mixing two varieties to get a better variety. For example, I will mix the red rose with the white rose to get a red and white colored rose. That is called hybridization. Okay. Now all of you answer this question. Self-pollinating plant versus cross-pollinating plant. Which plant is suitable for hybridization? In which type of plant you can easily make the hybrids? Yes, everybody? Okay, cross-pollinating plants. So can I say that the self-pollinating plants are not suitable for not suitable for hybridization. Do you agree? Self-pollinating plants are not suitable for hybridization. Now, okay, what about our natural mustard? Our natural mustard is self-pollinating. Okay, so all of you tell me, can we make hybrids with our natural mustard easily? Using natural mustard, can we make the hybrids? Yes? No. So therefore, not suitable for hybridization. Okay, therefore, not suitable for hybridization. So now what we should do? Okay, we should add some genes to change it into, yes, fill the blank. In the GM mustard, we should add some genes. We are adding the genes to change this self-pollinating plant into cross-pollinating. Is that clear? Yes. But how we can do that? Okay, how we can do that? Look at here. The first gene we are discussing here is Barnase gene. Use the name once. Barnase gene. Okay. So step 1. Barnase gene will silence the stamen. Is it clear? Means the mustard plant became male sterile. Is that clear? Like this. You have the stamen, you have the pistil. Okay, what the Barnase gene is doing? Barnase gene will inactivate, okay, inactivate the stamen. So it will make the plant male sterile. Is it completely sterile or only male sterile? it is only male sterile. Okay? Means pistil is active, female part is fertile. That is step 1. Is the step 1 clear? Add the Barnes gene. It will inactivate the stamen. Step 2. Okay. Add the pollen grain. Add the pollen grain from the variety you want. Is it clear? Okay. Means I want to add the pollen grain of a high yield variety. Okay, add it. I want to add the pollen grain from a better flavor variety. Add it. From whatever mustard variety you want, bring the pollen grain and add it. Is that clear? For easy understanding, I will just make a small comparison here. Let us compare this stamen as a possessive boyfriend. As long as the stamen is active, it will not let the pollen grain of any other plant fall on this. As long as this stamen is active, it will prefer to send its own pollen grain onto the pistil. It will not let the pollen grain of any other plant fall on this. So, but you want to add the pollen grain from some other plant. So what you have to do, give a small sleeping pill. Okay, this possessive boyfriend will sleep for some few hours. Meanwhile, you add the pollen grain that you wanted to add. Completed. Then step 3. Is it clear? Step 3, what we do is, the other gene, there is another gene here called as bar star gene. Bar star gene. What it will do? Okay, bar star will reactivate the stamen. Means it will restore the male fertility. Is it clear? Why? You don't want this plant to be completely sterile forever. Why you wanted to inactivate the stamen? You wanted to inactivate the stamen only to add the pollen grain that you wanted to add. Now I have added the pollen grain. My work is over. Okay. So the stamen can be reactivated so that the plant will be healthy and fertile again. Is it clear? Right? Clear till now? Perfectly okay. Your name says perfectly okay, so no problem. Okay. Right. So bar star reactivate the stamen to restore the male fertility. Is it clear? So now, is the hybridization completed meanwhile? Okay. What was the step 2? Step 2, we have added the pollen grain. Okay, at which step, can you tell me, choose between the 3, 1, 2, 3, at which step we have done the hybridization? Adding Barnes gene, or add pollen grain from the other plant, or add bar star gene? Which step actually reflect hybridization? The second step makes it hybrid. Very good. Second step makes it hybrid. First step made it GM, genetically modified, not hybrid. The third step also made it GM, genetically modified, not hybrid. Hybrid does not mean adding a gene. Hybrid means mixing two varieties. How did we mix the two varieties? By placing the pollen grain of one variety on the other variety's female plant. Is that clear? So the first and third step where we have added the genes will make the plant a hybrid plant or a GM mustard. Hybrid mustard or a GM mustard. First and third steps made it GM mustard. Second step made it hybrid mustard. So now this is a hybrid. It is both hybrid and genetically modified. We call this as DMH11, Dhara mustard hybrid 11. We call it as DMH11 or Dhara mustard hybrid 11. Okay. Then in this Barnase gene, and there is one more gene called Bar gene. Look at here, there are total 3 genes. There are total 3 genes. Okay. These 3 genes are taken from the Bacillus amyloliquefaciens. Earlier we have taken the BT gene from Bacillus thuringiensis, right? Here also similar procedure, but from a different bacterium we are taking. Earlier Bacillus thuringiensis, this time Bacillus amyloliquefaciens. And earlier we have taken Crivanase gene, only 1 gene, but here you are taking 3 genes, bar star, barnase, bar. What bar star will do? Okay, it will barnase, what barnase will do? Male sterile. What bar star will do? Restore the male fertility. What bar gene will do? Bar gene gives herbicide tolerance. Okay, so that even if you spray herbicide medicines, this plant will not die. Bar gene will give herbicide tolerant. Okay, so even if you spray the chemical herbicides, our mustard plants will not die. They are resistant to the herbicide called as glufosinate. Is it clear? Now, let us consolidate all these points. Okay, look at here. Barnase gene is added. Barnase gene which gives male sterility. Bar gene that gives herbicide, that is glufosinate tolerance. They both were inserted into Varuna variety. Is it clear? They both were inserted into the Varuna variety. And then bar star gene is inserted into the early Hera 2 variety. So they kept 2 genes in one variety, 1 gene in the other variety, and hybridized the 2. So you got a genetically modified and hybrid variety called DMH11. Is that clear? Clear till now? Surya, no Surya. Then, now, okay, now what are the concerns raised? What is the problem with Dhara mustard hybrid? I hope DMH11 is clear for all of you. What is the problem with DMH11? First one, there can be long-term ecological, social, economic consequences. Plus, spread of the herbicide tolerance gene to the other varieties by pollination. By pollination, this herbicide tolerance gene can spread it to the other plant varieties. Okay. Threat to biodiversity. How is it a threat to biodiversity? For example, if you have a high yield mustard variety, will the farmers prefer to use will the farmers prefer to cultivate low yield mustard variety? Given a choice, you will cultivate high yield variety or low yield variety. Given a choice, I will cultivate high yield variety only. So the low yield natural varieties will be neglected. They will be eventually lost. They can even become extinct. So it is a threat to biodiversity. Also a threat to food safety. If this mustard plant is giving more yield and a commercial benefit, why do I cultivate food crops? I will try to cultivate this one only. So food safety issues will come, food security, food shortage issues can come. So these are the concerns. And another concern is it can also destroy the honeybees because this is a high yield variety. It is a faster yield. So what happens? Fewer flowering days. Means the plant will have flowers on it only for few days. And honeybees survive on the flowers. So flowering days reduced. Because of this, honeybees will not get enough nectar. So it will decrease the bee population. And if just honeybees species become extinct, total ecosystem will collapse. You might have studied the importance of honeybees in the ecology subject, isn't it? Because they are the main pollinators. If honeybees become extinct, the total ecosystem will get disturbed. Is it clear? That is another problem. Next one. GM mustard require double the quantity of the fertilizer and water. It needs more fertilizer. Okay, means what? Unnecessary soil pollution. It needs more water. We don't want such crops. We want drought resistant crops that can grow with little water. Why do you want a variety that needs double the quantity of the water? We already have water scarcity on the earth, right? Next. The Supreme Court of India appointed a technical expert committee which in its 2013 report recommended total ban on herbicide tolerant varieties. Is it clear? Because if they are herbicide tolerant, then freely we spray more herbicides. It will lead to more soil pollution. I repeat, if my mustard variety is herbicide tolerant, I don't have a fear that the herbicide will kill my mustard. So I will spray more herbicides freely. It will lead to soil pollution. This is the reason why Supreme Court ordered for a complete ban on herbicide tolerant varieties. Clear till now? Then solve this question. With reference to genetically modified mustard developed in India, consider the following statements. GM mustard has a genes from a soil bacterium, yes. Bacillus amyloliquefaciens. That gives plant a property of pest resistance, no. Then which properties it will give? It will make it cross-pollinating and herbicide tolerance. Not pest resistant, herbicide tolerance. GM mustard has the genes that allow the plant to cross-pollinate and hybridize, correct. GM mustard has been developed jointly by IARI, no. Delhi University developed this. Is it clear? So 2 only is the right answer. Okay. Next topic is transgenic animals. Transgenic animals simply mean genetically modified animals. Let us take the examples. First example is cow. Even the natural cow gives milk. Milk contains a protein called casein protein. Casein gene produces casein protein. What is casein protein? This is milk protein. Casein protein is nothing but the milk protein. Okay. Now, do the natural cow contain the casein gene? Yes, natural cow also contains casein genes. But what we have done is we have added extra copies of the casein gene, so that this cow gives 13% more protein milk. Milk quantity will be the same, but the milk will have more protein. Okay, means you don't have to purchase a protein shake. You just drink this cow's milk, you will get extra protein. Is it clear? Then, second example is, okay, human casein gene. Human casein gene into the cow genome. Human casein gene into the cow genome. Then the cow gives human milk. Is it clear? Cow gives human milk. Why this is important? Sometimes some babies, for example, newborn baby, but unfortunately the mother passed away, or premature baby, mother did not start producing the milk. Then usually we depend on the formula milk, that is milk powder. But these milk powders are not good. Okay, but if you keep human casein gene into the cow, cow gives human milk. So therefore this milk can be used for such babies. So this is another experiment. Okay, next one is a transgenic fish that grows faster. Simple, every fish contains growth hormone gene. because every fish grows. But here what we kept extra copies of growth hormone gene. So this fish grows 6 times faster. So you get bigger fish, they weigh more. It's a commercial advantage to the farmers. Clear till now? Next, transgenic monkey that glows. Look at here, transgenic monkey that glows. So here what they do is, from the jellyfish, they take the green fluorescent. Green fluorescent protein gene, not protein. The gene is taken. Gene is a source of the protein, don't get confused. Okay, this gene is transferred into the monkey's embryo. Then this embryo implanted. This embryo implanted into the womb. Now the monkey started producing the green fluorescent protein, a glowing monkey, glowing mice, glowing kitten. Okay, this is another example for transgenic mice. Is it clear? Clear till now? Okay. So, next topic is cancer therapy. By the way, transgenic animals and GM crops also come under recombinant DNA technology or not? Yes or no? Transgenic animals. Jellyfish green fluorescent protein gene into the monkey. Bacterium, Bacillus amyloliquefaciens bacterium genes into the mustard plant. Can we call them as recombinant DNA? Yes, because we are mixing the DNA of different organisms here. Can we call this as genetic engineering? Yes, because we are dealing with the genes. You are editing, you are modifying, you are manipulating the genes. Okay? Right. Now the next topic is cancer and cancer therapy. To understand what is cancer, first of all, we should know what is a tumor. A tumor is nothing but extra cell growth. Is it clear? Unregulated cell growth. If the cells grow faster than the requirement, that leads to tumors. Tumors are two types, benign tumors and malignant tumors. Benign tumors and malignant tumors. Benign tumors are the tumors that cannot spread. Is it clear? They can't spread from one organ to the other organ. Malignant tumors can spread from one organ to the other organ. Okay. And only malignant tumors are called cancers. Benign tumors are simply called as tumors. They are not called as cancers. Is that clear? Then, in the human body, we constantly have two mechanisms. One is called as cell division. The other mechanism is called as apoptosis. Cell division increases the cell number. Apoptosis decreases the cell number. Why apoptosis decreases the cell number? Because apoptosis is nothing but programmed cell death. Apoptosis is nothing but programmed cell death. Now quickly tell me, do we get cancer if the cell division and apoptosis are perfectly coordinated? If the cell division increase, cell death also increase. If the cell division decrease, cell death also decrease. Like this, if they are coordinated, will it lead to cancer? Fast everybody? No. Then, if one of them increase, the other should decrease. That lead to cancer. Quickly tell me, increase in which one of these two leads to cancer? An increase in which one of these two leads to cancer? So, cancer can be caused either due to the increase in the rate of cell division or due to the decrease in the rate of the apoptosis. Increase in the rate of cell division or decrease in the rate of apoptosis. Then, we have some genes called proto-oncogenes. Use this term once. I mean, you can message me in the comment section. Proto-oncogene. Okay. Proto-oncogenes are the genes that regulate and coordinate these two. Who will maintain? Okay. Who will maintain the balance between cell division and apoptosis? Who maintains the balance between cell division and apoptosis? Proto-oncogenes. Is that clear? Who maintains the balance between cell division and apoptosis? Proto-oncogenes. So tell me, are the proto-oncogenes good for us or bad for us? Are the proto-oncogenes good for us or bad for us? First, they are good. Why? Because they prevent cancer. Because they prevent cancer. Okay. But if a proto-oncogene gets mutated, okay, then it will become oncogene. So what is an oncogene? These are the genes that cause cancer. Why do they cause cancer? Because they are the mutated proto-oncogene. Proto-oncogene is preventing the cancer. If that is mutated, can it prevent the cancer? No. So we get the cancer. So what is an oncogene? Oncogene is nothing but a mutated or a defective proto-oncogene is called as an oncogene. Clear till now? Is oncogene good or bad? This is bad because it causes cancer. It is bad because it causes cancer. Okay. So with this we understood what is cancer and what causes cancer. Now we should move on to the cancer treatment. What are the regularly used procedures to cure the cancer? Regularly we use chemotherapy, in which we use toxic chemicals to kill the cancer cells. We also use radiotherapy, in which we use high intensity radiation to burn the cancers. And we also do surgery. Okay. Depending on the type of the cancer, we choose any one of them or all of them sometimes, or any two of them sometimes. Okay. But our topic is not chemotherapy or radiotherapy now. Our topic is recent developments in the cancer treatment. Can we proceed? New developments in the cancer treatment. In this first one is CRISPR-Cas9 gene silencing. You all know that CRISPR-Cas9 is a gene editing tool. It can delete mutated gene. What is the mutated gene you want to delete here? In this context, which mutated gene you want to delete? Oncogene. And then in the gap add the correct gene. What is the correct gene that you want to add in this gap? Proto-oncogene. Is it clear? Add the correct gene. Right? Then, in which cell we do it here? In the somatic cells. So, it's a somatic gene therapy. It's a somatic gene therapy. Then you may ask me, ma'am, earlier you said somatic gene therapy is very difficult because of large number of somatic cells. How is it possible here? It is possible here. It is possible because we edit only the cancer cells, but not all cells of the body. So we have limited number of cells. So therefore it is possible. I am not saying it is easy, but possible. Is it clear? But is it possible for all the types of cancers? For example, can I do this using CRISPR-Cas9? Can I delete the oncogene in all the blood cells to cure blood cancer? Yes, everybody? Can we delete this gene in all the blood cells to cure blood cancer? No, because blood cells are dispersed throughout the body. So catching hold of the cancerous blood cells and deleting the oncogene is impossible. It means we can use it only for localized cancers, and also we use it on the leftover cancer cells. We use it only on the leftover cancer cells after chemotherapy or radiotherapy or surgery. Because if I delete the oncogene and keep the proto-oncogene, it will control the growth but it cannot kill the cells. Do you agree? Like in the case of a surgery, CRISPR-Cas9 is not like a knife to cut the cancer. It is not some toxic chemical to kill the cancer cells. It is not a high intensity radiation to burn the cancer cells. So it cannot kill the existing cancer cells. So first kill the existing cancer cells using the chemotherapy or radiotherapy or surgery. And then on the leftover cancer cells, if you do this, then further growth of the cancer can be controlled. Regrowth of the cancer can be controlled. Is that clear? Then second mechanism is RNA interference. In the RNA interference, what we will do? Okay, look at here. For example, imagine this is the DNA. On the DNA, you have oncogene. Okay, on the DNA you have some oncogene. What is it? It is oncogene. Now look at here. What is this? This is oncogene mRNA. This is oncogene mRNA. Its sequence will be what? U, A, opposite. Is it clear? G for C, C for G, T for A for U, not T, because it is RNA, right? T for A. Is that clear? Then this process is called as what? This process is called as transcription, isn't it? DNA to RNA conversion is called as transcription, right? Then next what should happen? What is the next step? Next, the oncogene mRNA should undergo translation to form the oncogenes. Protein will be produced, isn't it? But here what we do is, we are taking, this is called, this red color one is called as, okay, this red color one is called as, fine, I'll change the color. This is called as small interference RNA, means siRNA, or interfering RNA, or RNAi. What it will do is, Look at here. Now this pen can write. The pen can write. Okay. Now if I close the pen with a cap, can the pen write anymore? No. So I am comparing this mRNA with the pen, and I am comparing this small interference RNA with the cap. Is it clear? Means this RNA will bind like A for U, C for G, G for C, A for U for A for U. Okay, it will have complementary base pairing. Okay, like a cap it is bound. What it will do? It will prevent the translation. Why this is called small interference RNA or interfering RNA? Because this RNA, look at here, this RNA is interfering with the translation of this oncogene mRNA. It is interfering with its translation, not letting it undergo translation. Therefore, it is called interfering RNA or small interference RNA. Is it clear? So, finally, oncogene protein is not produced. Oncogene protein is not produced. Means what? Oncogene is silenced. Oncogene is silenced. Okay. Means what? Further growth. Further growth of cancer is prevented. Further growth of the cancer is prevented. Okay. Now remaining all points, it is just like the first one. Okay. Means this is also somatic gene therapy. This is also possible because we do this only with respect to the cancer cells. We don't do this in every cell. Here also small interference RNA cannot kill the existing cancer cells. This one also we do it on the leftover cancer cells. Here also we silence the oncogene only. But the only difference here is, okay, this is occurring at the DNA level. This is occurring at the RNA level. Remaining all points are same to same. Understood? Yes, clear for all of you? Okay. In the CRISPR-Cas9, you are directly cutting the oncogene. Here you are not touching the oncogene. Okay. You are dealing with the oncogene's mRNA. Then, once this interfering RNA binds, then what happens? Ergo-not 2 protein degrades this mRNA. Okay. Then ergo-not 2. Ergo-not 2 degrades this mRNA. Then what happens? There is no mRNA, so there is no translation. Oncogene protein is not produced. Oncogene is not expressed. So the cancer cells cannot grow further. Clear till now? Okay. And this is also called as antisense RNA technology. Why this is called antisense RNA technology? Anti means what? Opposite. Okay, opposite. Opposite to the sense. Sense means what? mRNA. Okay, check this small interference RNA. Is this RNA sequence exactly opposite to that of the mRNA sequence? U for A for U, C for G, G for C, A for U. Yes or no? Therefore, this RNA is also called as antisense RNA technology. Is it clear? We can also use this technology. This technology used for silencing any unwanted genes. Means not only oncogenes. Do we use antisense RNA technology only to cure cancer, or we can use the same technology to inactivate any unwanted gene you have in any organism? Got it? Do we use antisense RNA technology only to cure cancer, or it can also be used in the other organisms to prevent the translation of any other genes? It can be used in any organism to prevent the expression of any gene. Okay? Then now, immunotherapy for cancer. Immunotherapy for cancer. So here what they do is, okay, here we have some cells called as cytotoxic T lymphocytes. Is that clear? Cytotoxic T lymphocytes. What are they? They are killer T cells. They are killer T cells. What they do? They kill cancer cells. They can kill cancer cells. Okay? On these cancer cells, you have two structures called as PD1 and CTLA4. PD1 and CTLA4. CTLA4 and PD1. Okay? What are they? They are brakes on the killer T cells. Brakes on the killer T cells. Is it clear? Means they regulate these killer T cells. Right? Now, okay. Now let us compare. For easy understanding, I will give you a small comparison. Let us compare them. Okay, let us compare this with a dog. I am comparing the killer T cell with a dog. And I am comparing the CTLA4 as a leash. Leash with which I am holding the dog. Is that clear? Now, so now tell me, let us suppose you are going for a walk on a pleasant morning. You should hold your dog with a leash, or you should just leave it on the people like that? Yes, quickly please. On a pleasant morning you are going for a walk. You should hold your dog with a leash. Same way, when you are healthy, the killer T cells should have these brakes. Is the comparison clear? When you are healthy, when everything is perfect, these killer T cells must be regulated. They need these brakes on the killer T cells. Yes or no? We need these brakes on the killer T cells. Correct? Yes, fast please. When we are healthy, we need these brakes on the killer T cells. But, okay, on a pleasant day you are holding your dog with a leash. But suddenly somebody tried to attack on you. Your enemy came. Then what you have to do? Unleash the dog. What you have to do? Unleash the dog. So here same way, that enemy is a cancer cell. Okay? The enemy that we are speaking is cancer cell. Okay. Then what you have to do? You have to unleash it. Means here what? The brakes should be removed. Means here the brakes should be removed so that the T cells can kill the cancer cells. Is that clear? So, now what happened is, a scientist called James P. Allison produced an antibody for PD1. What it will do? It will destroy the PD1. Okay. Another scientist developed an antibody. What is this? This is an antibody for CTLA4. What it will do? It will destroy the CTLA4. Is that clear? Now, what happens? Okay, both the breaks are removed. Is it clear? PD1 antibody destroyed the PD1. CTLA4 antibody destroyed the CTLA4. Now the breaks are removed. It is like the dog is unleashed. Now that the dog is unleashed, it can go and attack on your enemy. Means these cytotoxic T lymphocytes can go and attack on the cancer cells and kill the cancer cells. Is that clear? This is called immunotherapy for cancer. Done? Is immunotherapy for cancer clear? Then, next one, CAR T cell therapy. And by the way, this immunotherapy for cancer won the Nobel Prize. James P. Allison produced antibody for PD1. Okay, CTLA4, sorry. Tasuku Honjo produced antibody for PD1. They both shared the Nobel Prize in Physiology for the year 2023 or 2024. Is that clear? Okay. Then you have CAR T cell therapy. CAR T cell therapy and the recent developments related to CAR T cell therapy I discussed in the phase 1. Okay. And I am giving you the link here. If you click on this link, in that you will get the phase 1 class in which I discussed about CAR T cell therapy and the current affairs related to that in detail. Okay. Then next one, stem cell therapy. Okay. In the stem cell therapy, what happens is, let us suppose, okay, you have let us suppose there is a blood cancer. Then what happens? We used chemotherapy. We used chemotherapy for the blood cancer. Then what happened? Okay, the blood cancer cells die. And it is okay. This is what we are expecting. Okay, complete focus and keep responding, please. But side effect is healthy blood cells also die, isn't it? Healthy blood cells also die, which is not okay, isn't it? Do you want healthy blood cells to die during the blood cancer treatment? Fast, quick, please. No. So, it is a side effect, but still it is okay. But another serious side effect, a serious side effect is even the bone marrow cells die. Why it is a serious side effect? Because if the bone marrow cells die, then the blood cells cannot even be regenerated. The blood cells cannot even be regenerated again. If the healthy blood cells die, it's a problem, but not a very serious problem because new blood cells will start coming. But if the bone marrow cells are killed as a side effect, then what happens? Even regeneration of the blood cells cannot happen properly. Is that clear? Okay. So now what you have to do? You have to replace the dead bone marrow cells. Do you agree? How to replace? Replace or regenerate? Replace or regenerate the dead bone marrow cells. How? Okay. By injecting, yes, fill this sentence. How can we replace or regenerate the dead bone marrow cells? Yes, all of you. How can we replace or regenerate the dead bone marrow cells? By injecting the stem cells. I hope you remember the stem cells can regenerate can produce other cell types, isn't it? So the stem cells can form the bone marrow cells here. Okay? I repeat, stem cells can form the other cells. So here if I inject the stem cells, they can form the bone marrow cells. So these new bone marrow cells will regenerate the dead bone marrow cells or replace the dead bone marrow cells. Okay. If the dead bone marrow cells are regenerated, then what happens? If the dead bone marrow cells are regenerated, okay, then we can form new blood cells again. You can form the new blood cells again. Is that clear? Then there is one previous year question. RNA interference technology has gained popularity in the last few years. Why? It is used in developing gene silencing therapies. Correct. I told you using RNA interference you can silence any gene, not only an oncogene. It can be used in developing therapies for the treatment of cancer. Yes, correct. It can be used to develop hormone replacement. No. It is gene silencing, not hormone replacement. It has nothing to do with hormones, not related to hormones. Is it clear? siRNA is not related to hormones. It can be used to produce crop plants that are resistant to the viral pathogens. Yes. How? By silencing the receptors. By silencing the receptors, viral receptors on the host cell. Okay, means what? Look at here. Imagine this is the host cell. On this you have a viral receptor. Then the virus will bind to this receptor. And like that, this viral receptor is like a gate. Where do we have the gate? Okay, in the host cell you have the gate. The virus will enter into the host cell using this gate. Now if that receptor gene is silenced, then the receptor will be, look at here, this is host cell. Host cell. No receptor. No receptor for virus. Because of that what happens? No viral infection. No viral infection. Clear till now? So like that, using RNA interference you can also develop plants that are resistant to the viral pathogens. This particular plant cannot get the viral infection. Any doubt till here? Then, next topic is DNA fingerprinting. DNA fingerprinting is also called as DNA testing or DNA profiling. Okay, because it is, now we are preparing for prelims. Procedure of the DNA fingerprinting is not needed. Procedure is not needed. So let us briefly see the procedure. Okay. Let us suppose there is a criminal case. Okay. So recently in the R.G. Kar Medical College case, a girl was raped. In her body they found the semen of some people. Okay. And then one particular boy was seen going out of the room in the CCTV camera footage. So what they will do is they will collect the DNA from that semen and will take, that is called crime scene sample. Is it clear? We take the crime scene sample compared to the suspected person's body sample. If it matches, the suspect became the criminal. The suspect is proven to be the criminal. If it does not match, no match, the semen found in her body, the DNA in that semen is not matching with the DNA of this man. It means what? The suspect is excluded from the case. Is that clear? The suspect is excluded from the case. Like this we can use it in the criminal cases. We can also use it. Okay, we can also use it in right, we can use it in criminal cases to identify the culprit sometimes, to identify the victim sometimes. Culprit, for example, as I told you, this R.G. Kar Medical College case. Okay. Victim, for example, that Shraddha Walkar's case. Okay. I'm unaware of the correct spelling of Shraddha Walkar. I'm sorry if I wrote the spelling wrong. Okay. So it means, for example, in that case, if you remember, a girl was cut into, her body was cut into multiple pieces and the pieces were thrown in different places. Now, how should you know whether these are the body remains of Shraddha Walkar only or not? Okay. So in that cases, like what we can do is, we compare it with, in the case of culprit, we compare the crime scene sample. The crime scene sample with the suspect sample. For victim identification, we compare the victim DNA with her parents. Means now take that DNA from those body remains, compare them with the parents of that girl. Unfortunately, if it matches, we should understand that this is the same girl who died. The body remains belong to that girl. We should understand that the body remains belong to that girl only. Is that clear? We can also use it to solve civil cases. Civil cases, for example, disputed paternity, cases. Like for example, a boy claims that so and so celebrity is my father, and the celebrity refuses and says that he is not my son. In that case, we can compare the DNA of the two. Is it clear? So father and the child DNA, 50% match. Then we can say that he is the father. Is that clear? We can also use it in the mass calamities cases. Mass calamities like Ahmedabad plane crash. In that you will have the boarded people's list. The body remains will be compared to the parents of the boarded people, the people who entered into the, who took the boarding pass and boarded into the flight. Okay, so their parents' DNA will be matched with the DNA of the body remains to identify a particular body remain belongs to which family or which parents. Is it clear? Okay. Then we can also use it for ART cases like surrogacy. For example, as I mentioned, Priyanka Chopra and Nick Jonas had a baby through surrogacy using a surrogate mother. In future, if that lady surrogate mother demands that this is my baby, can we solve it using the DNA testing? Yes. If we do the DNA testing, Okay, DNA will match with genetic parent or the biological parent? DNA will match with genetic parents or the biological parents? DNA matches with the genetic parents. So the surrogate mother cannot claim that this is my baby. So these are the various applications. And in the procedure, first what we do is we extract the DNA, then we amplify the DNA. Is it clear? We amplify the DNA, then we cut it into pieces, and the length of those pieces will be compared. If the length of the pieces match, okay, we will do gel electrophoresis. If the length of the pieces match, then we say that it is a perfect match, otherwise no. Okay, like for example, look at here. Okay, applications we already covered. Okay, analysis and comparison. Now procedure. Step 1, extract the DNA. Is that clear? Extract the DNA from the crime scene sample. Step 2, okay, amplify. Amplify means polymerase chain reaction. In an earlier topic, I told you polymerase chain reaction is nothing but in vitro DNA replication, isn't it? Replicating the DNA in the laboratory, isn't it? Then why we should amplify the DNA? Because in the crime scene we don't get sufficient amount of the DNA to do the test. So we should amplify the DNA and then only we can do the test. The third step what we do is we cut the DNA into fragments using, okay, restriction endonucleases. Using restriction endonucleases. Okay, what are they? Like Cas9, they are also molecular scissors. They are also molecular scissors. Is that clear? Then fourth step is called as RFLP. RFLP means restriction fragment length polymorphism. Restriction fragment length polymorphism means what? Difference in the length of the fragments. Okay, then what they do is, fifth step is called as gel electrophoresis. Gel electrophoresis. In the gel electrophoresis, what we do is, we do like this. Like for example, these are the 4 DNA fragments from A. These are the 4 DNA fragments from B. So 4 out of 4 matching. It means in the gel. See, for example, it is a gel. In this gel, we subject this gel to electricity. Electrophoresis means what? Okay. Electrophoresis means movement under the influence of electricity. So here what you do is, this is a gel. Label it as A and B. A and B are 2 people. I hope you are getting this. A and B are 2 people. Now 1, 2, 3, 4. Okay, 1, 2, 3, 4. subject this gel to electricity, means electrons are moving upward. Where the electrons are moving through the gel, electrons are moving like this. Imagine this cursor is an electron. Electron is moving like this. While the electron is moving, what is obstructing the electron? The DNA fragment. DNA fragments are obstructing it. So the moving electrons will push the DNA fragments upward. Now, tell me one small common sense answer. Okay, out of 1, 2, 3, 4, which one do you think is the smallest fragment? Out of 1, 2, 3, 4, which one do you think is the smallest DNA fragment? Yes? 4. So, fourth is smallest, therefore lightweight. So, electrons could move it to longer distance. Electrons could move it to a longer distance. Is that clear? So, from this, do you agree the position in the gel reflects the fragment's length, position in the gel reflects the length of the fragments? Yes, all of you? So, it means position is matching. Position matching means length matching. Position matching means length matching. And we are going to close this class in the next 15 minutes. Okay. So, complete focus and be alert, please. Okay. Position matching means same position means same matching. Okay, same length. Why? Because longer fragment. Okay. Acha, you said 4 is the smallest one. Wrong. 1 is the smallest one. Okay. 1 is the smallest one. Why 1 is the smallest one? Logic it is given here. Smallest, lightweight. So electrons could easily push it to a greater distance. So it travelled greater distance from the start point. Is that clear? This is the start point. So electrons could push it for a longer distance. 4 is heaviest. 4 is heaviest. So it could not move. It could not move. That is the reason why if it is present close to the start point, if it is close to the start point means longer. Little away from the start point means little shorter. Little shorter, shortest. Now look at here. Okay, between A and B. A and B are 2 people. A and B are 2 people. Between these 2 people, all the 4 DNA fragments are matching. So, it means 4 out of 4, 100% matching. Then what is the relation between A and B? What is the relation between A and B? %100% matching means AB are either same person or identical twins. How can it be same person? Probably this is the crime scene sample. This is the suspect sample. Okay. The DNA taken from her body plus the DNA taken from the suspect person's body. Is it clear? So those 2 are matching means he is the criminal. Or else identical twins also. If you remember, I said identical twins have identical DNA. There is one important limitation of the DNA fingerprinting. it cannot differentiate between identical twins. DNA testing cannot differentiate between identical twins. Between identical twins who did the crime, it cannot say. Okay, fine. Now answer this question. Let us suppose here out of 4 fragments 2 are matching, other 2 are not matching. So what is the relation between A and B? Only 2 out of 4, 50% matching. These are parent and child. Is it clear? Same way, if 75% matching, 3 are matching, 1 is not matching. 3 out of 4, 75%. It means they are siblings. A and B are siblings. Siblings have more matching when compared to the parents, because any one parent will give only 23 chromosomes to the child. Any one parent will give only 23 chromosomes to the child. So each parent can have only 50% matching. But siblings have both the DNA, siblings have both the parents common. Is it clear? So therefore, between parent and child 50%, but between 2 siblings you can have above 50% matching. 75 is not fixed, but above 50% means they are siblings. Okay? Above 50% means they are siblings. Okay? 25% matching, there can be some relation, but we can't say exactly what is the relation. This is called DNA testing, DNA profiling, DNA barcoding, DNA fingerprinting. By the way, why this is called DNA fingerprinting? Is there anything related to the thumb impressions here? No. Here fingerprint word symbolically represent that this pattern is unique. Is it clear? This band pattern, the band pattern is unique like a barcode. The band pattern is unique like a barcode. Okay. So no 2 people have the same band pattern. So therefore it is called DNA fingerprinting. That band pattern is also called DNA profile. DNA profile is nothing but that band pattern. Okay. Then next one is DNA Technology Use and Regulation Bill. It was introduced in Lok Sabha in 2019. It seeks to establish national data bank and regional data banks. National and regional data banks of what? National and regional data banks of DNA profiles. What is the advantage? Easy identification of the people. What is the limitation or what is the criticism? Right to privacy. Okay, right to privacy. Is it clear? Okay. So it means I can say I will not give my DNA because my DNA will reveal sensitive information about me. My DNA reveals sensitive information about me. So I have a right to refuse to give my DNA. Is it clear? Okay. Thank you so much. Then consider the following statements. DNA barcoding can be a tool to assess the age of a plant or animal? Wrong. We cannot use this to assess the age. Why we cannot use to assess the age? Because DNA sequence do not change with age. Whatever DNA you had when you are born, same DNA you will have on the last day also. 90-year-old also will have the same DNA sequence. Minor changes can be there because of the mutations, but DNA testing cannot anyway detect them. Okay. Distinguish between the species? Yes, because different species have different DNA sequences. Identify undesirable animal or plant material in the processed food. You know something, fun fact, mutations will be there, but that is very rare. Mutations are rare. Okay. Amar, with age we can get mutations. So there can be slight change in the DNA sequence. But such minor changes, anyway the DNA testing cannot recognize. So there are no considerable changes in the DNA sequence with age. Using DNA we cannot say the age of a person. Is it clear? Next. Undesirable plant or animal material. You know one point, fun fact, in coffee powder, chocolates and all, some cockroach material is permissible. It means if you have certain amount of cockroach material, okay, it is not illegal, like it means nobody will ban that particular chocolate or that coffee powder and all. It means what? Unavoidably some cockroach material goes into these food products. Is it clear? Then how to know if it is contaminated or not? You get your coffee powder. In that, if you have coffee plant DNA should be there. But if the coffee powder contain cockroach DNA, what does it mean? The coffee powder is contaminated with cockroach. So like that, DNA fingerprinting can be used to identify the contaminating plant or animal material in the food products. Is it clear? Yes, that is also correct. 2 and 3 is the right answer, right? Okay. Next question. With reference to recent developments in science, which of the following statements is not correct? Pieces of artificial functional DNA can be created? Yes. Why only pieces? Complete genome also can be synthesized. HGP, right? Complete genome also can be synthesized. HGP, right? So of course, okay, pieces of artificial functional DNA can be manufactured. Okay, then a piece of DNA taken out from an animal cell and can be made into replicate outside a living cell? Yes. Can you tell me what is this procedure called? DNA taken and this DNA is replicated outside a living cell, not inside a living cell. Yes, anybody? A DNA segment is taken? No. CRISPR-Cas9 will cut the DNA. It will not replicate the DNA. Replicate means copy. No. Recombinant DNA technology, correct, but that will copy the gene inside the living cell. But it is asking for outside a living cell. Polymerase chain reaction. In vitro DNA replication. Okay. Cells taken out from the plants and animals can be made to undergo cell division in the laboratory petri dish. Yes, it is called tissue culture. It is called tissue culture, and yes, we can grow them like that. Okay. Then, now, lastly we have infections. In the infections we have bacterial diseases, viral diseases, fungal diseases and protozoal diseases. Here I have given a list of bacterial diseases. Okay, nothing to explain in this. Name of the disease, I will tell you one example. Name of the disease, tuberculosis. Caused by the bacterium, Mycobacterium tuberculosis. Spreads through air. And the main system affected is respiratory system will get, which part of the body will get affected? Respiratory system will get affected. Can you go through this table like this? Nothing to explain, it is a factual content. Done? Okay. Then you have viral infections. First, what is a virus? What is a virus? A virus will have two components. Next 5 minutes complete focus please. 5 minutes absolute focus. Virus will have a protein coat and the genetic material inside. Protein coat and the genetic material inside. Now, okay, if the genetic material is DNA, then we call it as a DNA virus. Is that clear? Then, if the genetic material is RNA, then we call it as RNA virus. Is that clear? Third type, if the genetic material is, listen this point very carefully please, last point that I will explain, okay. I am saying as if your mother say like one more piece you please eat, okay, like that. One last point I will explain, okay. If it is RNA but it will be converted into DNA, it will be converted into the viral DNA, right? Then it will insert the viral DNA into the host cell DNA. That is called retrovirus. Okay, I repeat, it will have RNA in the genetic material, but it can convert its RNA into DNA. Then it will insert its DNA into my DNA. That is called retrovirus. Why that is called retrovirus? Because you all know that DNA to RNA is called transcription. But RNA to DNA is called reverse transcription. It is a retro process, isn't it? It is a retro process. Retro process means what? Backward process. Retro process means backward process. Okay. Therefore, a virus which can do this is called retrovirus. Okay. It will have RNA as genetic. This is its genetic material. But its genetic material, for example, HIV RNA will become HIV DNA in the presence of an enzyme called as reverse transcriptase. Then the HIV DNA gets inserted into the human DNA. Is that clear? HIV RNA will get converted into HIV DNA with the help of reverse transcriptase, and the HIV DNA will get inserted into the human cell DNA. This type of viruses are called retroviruses. I have given examples for all. HIV, human T lymphotropic virus, they are the retroviruses. RNA viruses examples, most of the human viruses are animal viruses. DNA viruses examples, herpes, smallpox, adenovirus, hepatitis B, human papilloma virus, these are the DNA viruses. Is that clear? Lastly, you have some viral infections list, you can go through this. It's a factual content. Okay, name of the disease, name of the virus, how it is transmitted by sneezing, coughing like that, which body system gets affected. And then lastly you have protozoal infections. What are protozoa? They are unicellular eukaryotic cells like amoeba. One recent example is brain-eating amoeba. Brain-eating amoeba called as Naegleria fowleri. Naegleria fowleri. Is it clear? So that is called brain-eating amoeba. You know, amoebas are protozoa. Protozoa are eukaryotic. Eukaryotic means what? They have a nucleus. They have a nucleus. Eukaryotic unicellular organisms that cause infections. Example, brain-eating amoeba. Our malaria parasite, Plasmodium, is a protozoa. It causes malaria, spreads through Anopheles mosquito. Leishmania is a protozoa. It causes leishmaniasis disease, spreads through sandfly. Trypanosoma is a protozoa. It causes trypanosomiasis, spreads through CC fly. Okay, Entamoeba histolytica is a protozoa. It causes amoebic dysentery, spreads through contaminated food and water. This is a list of some protozoal infections. Is that clear? Okay. Then after that we have vaccines and all we have already discussed. Nuclear technology we will discuss in the next class. So in today's class, part 1 I completed. In part 1 I completed biotechnology. Part 2 will be there on March 13th, where I will discuss nuclear, ICT and nanotechnology. Part 3 will be there in the late March, March ending. Where I will discuss space technology and defense technology. Okay. So I will cover the total science and technology if you just attend these 3 classes. Okay. So that's all for all of you. Thank you so much. And if you like this class, if you found this class useful, do let me know in the comment section. And don't forget to subscribe to the channel. Thank you so much. All the very best.